Good afternoon and welcome back. Good morning, depending on where you are, and welcome back to day 2 of our public meeting on understanding priorities for the use of digital health technologies to support clinical trials for drug development and review. I'm Mark McClellan, the director of the Duke-Margolis Center for Health Policy at Duke University, and I'm delighted to be opening day 2 of this important and timely meeting. As a reminder, we're holding this event in part to meet a PDUFA 7 commitment for the FDA, holding a series of 5 public meetings on the use of digital health technologies, or DHTs. Yesterday, we had a wide-ranging discussion that covered a whole set of issues on how DHTs are being used in clinical trials and the opportunities, challenges, and lived experiences about how DHTs can potentially impact the diversity and accessibility and effectiveness of clinical trials. We covered a lot of ground in that discussion. Just to recap a few of the key points, one is that it's essential to make sure that patients and their caregivers and family members are centered in the development and application of these tools, their integration into clinical trials for drug development. Filling gaps in meeting patients' need, that's a prime motivator for this work on DHTs. Second, there's an enormous potential in this space. And even though there's some concerns and things to watch out for, stakeholders across the board are seeing a lot of promise from DHTs that is increasingly borne out in the data and the clinical trial experience. We also heard about the importance of reaching out to people to make sure that access is offered and the approach to potential participants needs to be personalized so the trials are accessible and diverse and work for a broad range of populations. Today, we're going to go deeper into how some specific sensor-based DHTs are already being used. That includes actigraphy, continuous glucose monitors, remote cardiac monitors, and environmental sensors. We're going to use these examples to hear about use cases for these technologies and flesh out some of the discussions that we had yesterday. We'll then conclude the meeting with a session that aims to be forward-looking regarding where the key opportunities for the advancement and integration of DHTs into clinical trials to support drug development are, based on the discussions over the last couple of days. Now, just like yesterday, we want everyone who's joining us today to share their thoughts, comments, and any questions over Zoom. We're going to try to handle as many of those questions as we can. Our team will review the questions and comments as they come in. We'll route them to our session moderators and to other participants on our panel so that the points or questions raised in Zoom can be put into the panel discussions. In some cases, we may be able to answer them directly in writing. So for all of you who are with us today, if you would like to ask a question during the presentation and open discussion portions of the meetings, just type it into the Zoom Q&A box if you're logged in through Zoom. If it's like yesterday, we may get a large volume of comments and questions. We may not get to all of them, but we'll try to get to as many as possible, and certainly helpful for the records from this meeting as well. So please keep those questions and comments coming. We'll do our best to get them all incorporated into the discussion today, both among our panelists and online. So let's get started. We have a lot of ground to cover today. I'm very pleased to introduce Dr. Christina Silcox, who is our research director for digital health at the Duke-Margolis Center. She'll be moderating session 3. Christina?
Thanks, Mark. Hello, everyone, and welcome to session 3, Actigraphy and Clinical Trials to Support Drug Development. In this session, we'll be exploring how actigraphy is being used as an outcome measure to assess different medical conditions and different kinds of patient populations, and how their use has changed clinical trials. During this session, we're going to be using the term actigraphy in the broadest sense to mean any accelerometer-based measure. And these presentations will cover what actigraphy is and how it's measured, as well as different use cases for actigraphy as an outcome measure and what the patient experience is like using actigraphy-based DHTs in clinical trials. So to start, I'm going to hand it off to Jeremy Wyatt. He's the CEO of Actigraph. Jeremy?
Thank you, Christina. and thank you for everybody letting me join this great panel. You can go to the next slide there, Christina. Thank you. My name is Jeremy Wyatt, and as Christina said, I'm the CEO of Actigraphy, and we are a medical grade wearable and digital biomarker company, and our mission is pioneering the digital transformation in clinical research. I want to thank the FDA for this great workshop, and especially for this framework. This is a really important time in the history of drug development. So I'm going to talk today about actigraphy, and I'm going to try to get through this pretty quickly, so you all need to listen pretty fast. All right, next slide. There's a safe harbor statement. So let's go ahead and go to the next slide, Christina. So let's just talk real quick about what is actigraphy. I thought that Dr. Leonard Sachs yesterday did a great job of introducing this concept, but just to revisit it in a little bit more detail, essentially actigraphy is a non-invasive method of monitoring human activity in rest cycles. And so it's as basic as that. It's typically done with a small device like the one we're showing here on the screen, and that device is typically worn on the body, either on the wrist or ankle or thigh or waist over a period of days or weeks. The primary sensor that's used around actigraphy is an accelerometer. That's sort of the core technology that's in there. So there's a difference between accelerometry and actigraphy, and we'll look at that today. There's other components in here like battery and memory for storage, and then connectivity that allows you to get the data off of the device. Typically what happens is that data is captured, and then in some process, form, or fashion, that data is then interpreted using an algorithm or some method into some specific measurement like sleep or physical activity or caloric expenditure. And the graph on the right here shows an example of how sleep cycles are measured using actigraphy. Next slide, please. So I just want to make the point briefly that this is not new technology. This technology has actually been around in use in clinical all the way back into the 50s. These two papers here that I'm referencing are in PubMed, and they're older papers, but the picture on the right shows the publication from the 1970s where an older actigraphy device was used in the monitoring of sleep. And you can see we've come a long way with the technology. This was a mechanical-based accelerometer, and you can see it's attached to that person's body using a wire and a battery. It wasn't very patient-friendly. Fortunately, we've come a long way since then. Next slide, Christina. So real quick lesson here on what is an accelerometer. I thought I would just share this briefly because it is the foundation and the basis of actigraphy. And I know that Steve is going to present next and talk a little bit more about this in detail. So I won't spend a whole lot of time here, but these accelerometers that you see they're everywhere. They're in our phones, they're in our watches, our smartwatches, they're in our vehicles, and they're used to do a variety of different things. But a lot of folks don't understand that they're actually, whoops, we lost our slide there. These accelerometers are actually mechanical devices. And in fact, you hear the term MEMS, it actually stands for micro electromechanical system. And if you look on the left picture there, what you're seeing is an electron microscope view of an accelerometer, which actually contains a number of different masses that move against gravity. And so these capacitive plates move along with that mass. And from that, we're able to discern acceleration. And from your college days, most of you probably remember that acceleration is a change in velocity over time. And so it's a little mechanical system that's running in these accelerometers. Actigraphy is the approach where we use the accelerometer to measure things from that data. Next slide, please. So real quick, if let's take a look at this graph here. But if you don't take anything away from my discussion today, I want you to remember these two terms: garbage in equals garbage out. Before we even talk about human measurement, we have to start here. Technical verification is a fundamental requirement if we're going to use accelerometry to measure patient behaviors and patient movement. So accelerometers don't produce consistent measures out of the box. If I buy 100 of those that you saw on the last slide and put them on my desk here and hoped that each one would measure 1 unit of gravity, I would be wrong. Some may be 1G, some may be 0.97, some might be 1.04. And that doesn't sound like a lot, but when we're talking about consistency in clinical trials, it absolutely matters. The shaker table that you see on the left is a large orbital shaker that allows someone who's calibrating an actigraphy product to move that device in a plane so that it can experience different forces of gravity. And from that, we can characterize things like the offset that I just discussed and how we calibrate that. We can also characterize the signal drift. So what happens to the baseline sensor over time? And then lastly, the dynamic response. What happens when we actually experience 3, 4, 5, 6 Gs of acceleration? Sometimes these accelerometers, they may read, 3 Gs, but in fact, it's experiencing more. And typically these tables are used to calibrate these accelerometers against the NIST accelerometer. And in this example, Actigraphy collaborated with the NIH to develop a verification process as part of the NHANES study about 10 years ago. Next slide, please. So let's talk about the high fidelity raw accelerometry data in an actual accelerometer. If you can click the next part there, Christina. When we talk about raw data, we're talking about the actual measurement of acceleration directly off the accelerometer. And it's typically quite noisy. I mean, you can imagine the vibration in a car if you're riding in a car, what an accelerometer sees. Next slide, please, Christina. So the raw data is typically turned into a processed outcome. This happens after the technical verification because remember, garbage in, garbage out. We've got to be certain of the raw data's consistency before we can process the data. When we talk about processed data, we're talking about any measure that is derived from the raw data through some transformation process. Most of the time we call this an algorithm, but in fact, what's happening is that we're reducing the raw data into a form that allows it to be interpreted into a measurement that we're all used to. And you've seen these measurements on the right, calorie burn or sleep efficiency or step count. So that process allows us to take the raw accelerometry data and reduce it down to something that we can measure using some processes of filtering, aggregating, regression, and in some cases, artificial intelligence and machine learning. The point here is that you can't go backwards. You can't go from a processed outcome to the raw data. Next slide, please. We typically see two approaches in measurement in clinical trials. And so, Christina, if you'll fade the first in here. The consumer model, we see a lot of times in consumer products where we have a product that is measuring accelerometry and movement. And on that product itself, whether it's a watch or some type of wearable or off-body sensor, there's an algorithm that's applied to that data to turn those raw data into some outcome that we all are used to, like in this case, sleep efficiency. Next click there, Christina. But in the clinical model, what we prefer to see is that the raw data and the algorithms are actually separated. This allows a lot of benefits. When we collect just the raw data on the wearable, we're able to get very precise with that data and consistent from device to device. It also allows us to exploit things like a longer battery life, and it also reduces risk around collecting this data. That allows us then to use that data to interpret it in a number of different ways that might be specific to a population. For example, we might want to use the data we've collected to look at scratch detection on atopic dermatitis population. But we might find after that study that we need to change some things about that algorithm. If we didn't use raw data, we'd have to go back and collect that again. So in a lot of cases in clinical trials, we'll see that these are separated because it maximizes the flexibility and also gives traceability around how the outcome was derived and transparency around which algorithms were used to get there. Next slide. We see actigraphy today used in clinical research in a myriad of ways. For continuous data collection, not just episodic data collection, it provides a better picture of what's going on in the patient's real life. We see it used as a surrogate endpoint for better measures of efficacy of treatment. We see it used to improve engagement with patients, and this often leads to better adherence and retention when patients feel like they're part of something. And in the panel today, we'll talk a little bit more about that. And then lastly, we heard yesterday on the panel on DHTs on clinical trial accessibility and diversity, led by Dr. Lola Basho-Inajay. She did a great job yesterday of facilitating that panel and talking about broader access for patients in diverse backgrounds. And this really is an opportunity for us to enable greater clinical trials, broader clinical trials with better data. Next slide, please. So what do I mean by better data coming from an accelerometer or an actigraphy product? In a typical clinical trial, in this example, maybe there are 365 days in a year, and maybe we measure 5 different things that are happening with that patient throughout the duration of a trial. It doesn't really give us a good picture into what's happening in the patient's life. we might envision that these vertical graphs that you see here are some measurement of a 6-minute walk test or perhaps some in-clinic measurement. We see some trend, but what happened at the end? We're not sure. And so in reality, what's happening, if you'll click the next piece, is that the patient's functioning is improving over time, but we wouldn't know that if we didn't have an actigraphy product. And this is actual data from a study. So in the past, we've not been able to measure this, but technology has improved to the point where we can do this very consistently. And you might envision a patient who is relying on this drug in a trial or really counting on a therapy getting excluded from a trial, maybe because they didn't perform on that last portion of the trial measurement. So it's better for the patients and it's better for the clinicians. Next slide. So we see also that actigraphy just offers a better approach to clinical outcomes in many indications. So the first bullet here, actigraphy presents new ways to measure behaviors where gold standards do not exist or are insufficient. Good example is Duchenne's muscular dystrophy. I remember when Jen Goldsack started the Digital Medicine Society, she used this example of Duchenne's muscular dystrophy. The primary endpoint is 6-minute walk test. And many of these patients, after a period of time, their disease progresses, they end up in a wheelchair. And so this is a completely meaningless measurement for these patients. Second, I'll just remind you that FDA defines clinical benefit as how a patient feels, functions, and survives. And historically, the feel and function, specifically the function piece, has been very difficult to measure in the real world directly. But today, we have technology that can enable us to do that. In addition, these continuous actigraphy measurements that we see happening on these patients are far less burdensome on them. And at the same time, they produce better evidence. And then lastly, treatment and clinical benefit is the most challenging but essential step for drug development. And we can see some examples here with drugs like Adelum and Tofersen and others that really maybe the drug itself it wasn't an issue where the drug didn't perform up to standards, but rather we didn't have great tools to measure a treatment response. And because of that, we weren't able to get these drugs to patients as quickly as we needed to. And so we see the ability of actigraphy to really enable that and make that happen in a much faster way. And in fact, we've recently seen how new digital measures can do that. If you go to the next slide, I'll give an example. This is, there we go. In this example, MVPA is a measurement from actigraphy, moderate to vigorous physical activity, and it's being used as a primary endpoint in a pivotal phase 3. In this example, oxygen saturation and the 6-minute walk tests were conventional endpoints, but they did not show treatment effect. But the digital measure of MVPA did show clinically significant changes. The graph on the left shows the treatment response to the treatment group with an increase in physical activity in the maroon portion, and then a decrease in the placebo group using the same measurement. So the FDA did endorse this endpoint in a pivotal phase 3, and it's actually being used today in the measurement of this treatment. Also, because of the statistical efficiency of the measure, the sample size was reduced by 50%, which accelerated the development timeline and significantly reduced the cost of their phase 3 trial. So hopefully you got a little bit of a picture today around what is actigraphy, how can it be used, and what measurements might it replace in clinical research. And I think that's the last slide. Great. Thank you, Christina.
Jeremy, thank you so much for that presentation. Really interesting, and particularly happy that you had talked about some of those raw data measures and the importance of calibration and technical verification of kind of that basic accelerometer. I think that's a point that really came up a lot yesterday, particularly around some of the challenges around bring your own device, as well as some of the versioning control type things that happened. And I also really appreciate kind of you talking about this is a way to actually measure feel and function, which has traditionally been a difficult way of thinking about this. Our next presenter is Dr. Steven Zhu, who is the Assistant Professor of Dermatology at Northwestern University. So Steve, I will turn it over to you.
Thank you so much. Next slide. So really excited, Jeremy. Great talk and a great segue into what we're going to talk about, which is actigraphy and beyond, and how we're using some of our technology for novel digital endpoints for clinical trials. I have two affiliations. One is Northwestern, the other is at Cybal Health. where I'm currently CEO and co-founder. Next slide. This is my disclosure. Next slide. So I'm a huge fan of Dom, everything that they do. And, I love this. I modified a little bit, but basically, as Jeremy was saying, the ability to take some of these sensors home can really reveal both objective measures, but continuous data that really reveals the true patient experience outside of what we see as physicians in the hospital setting, both in clinical trials and clinical care. Next slide. And so I was very fortunate to be a participant in this awesome initiative that we heard about from day one. And it was just really great to see in a pre-competitive way, so many different partners that ultimately care about bringing this endpoint forward to support drug development. It was special for me as a dermatologist to be able to see that happen in my field, where I take care of so many patients with atopic dermatitis directly. Next slide. So I want to talk a little bit about our work and what we've done. So, risk-based actigraphy is really powerful, as Jeremy's saying. You use a lot with it in terms of understanding the condition, and it's actually been tested in the context of scratch sensing as a potential surrogate marker for itch. So most of that work has been done with actigraphy and wristbound systems. Next slide. And for those of you who struggle with insomnia, you can read this review paper we actually put together just a year ago around some of the work that's been done in this space. And I think suffice to say, our take home was actigraphy is used the most, but the performance of the algorithm really depended heavily on whether you tested it in healthy normals or the actual context of use in AD patients. So I think that really underscores how important it is to validate these technologies in the actual context of use, because the results were very different, at least in our review. Next slide. So how are we doing things a little differently? So I would describe what we do is advanced wearables that are skin mounted, and we're doing something called high frequency actigraphy. So we're taking kind of accelerometers, the same thing Jeremy was talking about, but we're picking ones that have high frequency sampling that kind of get into the acoustic range. So ADAM is actually short for advanced acoustomechanic sensor, and it gives you sort of interesting information that may be missed if you're just doing gross motion at lower sampling rates. Of course, that comes at a cost in terms of memory and battery life, but we think it's relevant and useful for other applications, especially for some of these novel digital endpoints. Next slide. So our work here, if you can press click once, you can see this video. And basically what we've done is we hand mount our sensor on the dorsum of the hand, embed an AI algorithm leveraging edge computing. And when that sort of happens, you can sort of see scratching events detected. If you click the next slide, just click once, and the bottom video too, if you will. Basically what we did is we trained our algorithm and sensor on healthy normals and then actually applied that to AD patients, both kids and adults. So some of these are my patients actually, and you can see infrared camera while they're sleeping wearing our sensor, being able to label that against it. So all that work translating into basically validation of these technologies for the context of scratch. Next slide. And so we published a paper in Science Advances. If you click the next slide. And what we illustrated is that basically when you mount the sensor on the dorsum of the hand, you're actually capturing finger-based motion. And if you can see on my video, my wrist is largely motionless, right? So that's kind of one useful thing about applying this. But then also it's more specific because getting that high frequency signal, we can differentiate from motions that look exactly like scratch, but be able to not sound like it. So I think that's been kind of an interesting opportunity where we're taking actigraphy to a high frequency state where we can classify scratch with more accuracy. Next slide. So if you just click through, so the whole animation plays. So we validated it in 2 journal articles. It was great to see the popular media attention. I just want to send out an appreciation to the FDA. We're part of the drug discovery tool program. I think we're one of 6 kind of digital health technology, passive technology monitoring systems accepted into the DDT program. So it's been a great, process working with the FDA through that. And also thank you for the grant support in developing this as an endpoint. Next slide. And so in a follow-on paper, what we did was we really wanted to link scratch and sleep. So if you think about atopic dermatitis, which is kind of the hallmark disease where itch is the major symptom, what we're also showing here is that based on investigator global scale, the severity of the disease is actually associated with increased scratch events per sleep hour, captured by our sensor. And the other thing that we sort of noticed is that we also can capture an intensity score with our scratching. And that also seemed to get worse with worsening global assessment of the disease. So I think it's great to sort of see that being linked to actual like disease manifestation. And we published that in the JAD just last year. Next slide. What we also did was link on the same sensor. So we're using actigraphy to look at sleep stages and sleep time, that scratch events and sleep time were inversely related, which makes a lot of sense. The more you're scratching, the less you're sleeping. So sort of great to see that kind of manifest as well, linking scratch and sleep together. And then conversely, what's directly related is kind of wakefulness or awakenings after sleep onset increases with the number of scratch events per hour. So I think it's just sort of cool to see these things kind of connect together into concepts of interest that really matter to the patient, which is itch and sleep. Next slide. And so we're moving forward beyond nocturnal scratch to think about daytime as well. We think that there's a lot of interest in understanding itch throughout the context of the daytime and nighttime. Some patients endorse much worsening itch at night. So we're actually exploring daytime itch as the sort of next step of our innovation cycle, is to train our system to detect itch no matter whether you're doing different kinds of activities, no matter where you're scratching on the body. Next slide. And that includes creating new systems where we're looking at, making sure things like texting or typing or even brushing with an electronic toothbrush is correctly identified as not scratch, so that we can kind of be able to provide daytime. Next slide. And then can you play the video quickly? And so one of the things that we're always thinking about is how do we do more for patients? I think monitoring and measurement is very important, very useful, especially for clinical trials. But we also think about clinical care. Like how can these be adopted beyond just clinical trials? And so what we've actually done is embedded a haptic motor in our sensor to actually vibrate when it detects scratching. And this is fully closed loop. You don't need a mobile phone. You don't need a watch. You don't need an app. Basically, the sensor itself is detecting it. And some of our patients are finding this really useful to get them to stop subconsciously scratching. And the idea here is this might be useful beyond monitoring to help as an adjuvant, for instance, to a drug or a topical or therapy, kind of thinking about how this can do more than simply an outcome tool. Next slide. Next slide. You can skip the video. You don't have to play the whole thing. And so beyond atopic dermatitis, taking this sensor, we're really excited about taking it from the hand, actually putting it in novel locations and be able to do other digital biomarkers together. Next slide. So with the same scratch sensor, we're using it to look at swallow. And Michael J. Fox Foundation is a partner, a grant body agency. We're working with us. We already submitted it for consideration to the FDA as a new digital endpoint for swallow. And we're using the same thing for cough, just taking it from the wrist, putting it here and be able to detect these kind of novel biodigital markers at the same time. And then last slide. And ultimately, I think our vision is how do we take that, prove it in these indications, these contexts of use, put it in peer-reviewed publications, and then ultimately take this from clinical trials to clinical care. That's kind of our vision, that tools that are useful in the context of measurement, I think has a future potentially to also support the entire sort of disease and entire patient with a drug after it's approved. So thank you very much for your attention.
Thanks, Steve. Great presentation. And I think you really made a lot of really important points, particularly around being able to do the work to do differentiation. I know my father always laughs when he gets his 10,000 step count while he's brushing his teeth at the end of the night. I think also, the way that we can customize these things for different applications, I think was really interesting, and the ability to do some differentiation. During the panel discussion, I'm definitely going to want to talk a little bit about how patients, what patients' experience with wearing some of these devices was, as well as I was really interested in hearing about giving that alert feedback and any concerns there may be around how that could affect the clinical trial results. So lots of stuff to talk about, but we are going to move on to the next presentation, who is Dr. Diane Stevenson. She is the Executive Director of the Critical Path for Parkinson's Consortium at the Critical Path Institute. So Diane, I will turn it over to you.
Hi, Christina. Thank you so much. It's such an honor to be in this session. And I'm so pleased to represent Critical Path Institute, a nonprofit organization whose mission is to improve human health. We improve the health of patients and populations worldwide by accelerating the development of safe and effective medical products. If you flip to the next slide, and I want to acknowledge and thank the FDA for their generous support in both launching and the continued success of Critical Path Institute. The focus of today is going to be on the Critical Path for Parkinson's Consortium. Next slide. And this initiative really is so timely and urgently important. This is a disorder that is the fastest growing brain disease of urgent need of improved therapies. The current medications are not effective in meeting the long-term needs and disability and progression of this relentless disease. We were founded in 2015, thanks to generous funding from Parkinson's UK, and now have this incredibly exciting, engaged group of worldwide leaders collaborating together to accelerate treatment for patients. The failure rate of trials in Parkinson's is so high for many different reasons, but the pipeline is very rich with very promising therapies that target the underlying pathophysiology of the disease. So what a great time to collaborate together. And today I'll be talking about our 3DT digital drug development tool team, which was launched after we got CPP off the ground. Next slide. Thanks to tremendous progress in the field that you've heard about already at this meeting, the problem statement is great. We want to intervene as early as we can, but current outcome measures are not sensitive to identification of early impairments and functional change over time. This field is evolving fast, and thanks to the FDA for this exciting meeting, tremendous progress is being made, but we're not where we need to be, and we need to define ways for regulatory success in completely new, unprecedented ways. These exploratory tools are critical, but we need to share data and learn together. So the goal of this 3DT initiative is to advance a device-agnostic regulatory strategy for acceptance and approval of digital tools in clinical trials, targeting particularly early-stage disease. Next slide. So many people to thank. Our industry co-lead, Josh Cosman, who was a key catalyst to starting this initiative, and you'll hear about the case study today, told me many times in many companies when they go trying to do this on their own for the first time ever, they think it's pretty simple. If you look at these icons, pretty simple problem, right? You take a digital device tool that you choose and you put it in a trial, and lo and behold, you get data. But it's way more complicated than that. And if you try to find these two icons in the next slide, you'll see they're buried in a sea of, next slide, incredibly complicated diagram from a published paper that we have. When you try to do this on your own, you don't even know how complicated it is until you start dealing with all the complexities and complex factors that one needs to consider up front. And the hard thing is, for those who have been paving this path for some time, many of these things you don't even know until after you start dealing with the volumes of data, the security issues, the patient compliance, missing data, all these things. So what a great opportunity to collaborate together. So today I'm going to share with you a case example that this consortium has really been leading. Next slide. This all started thanks to some pioneering leaders at University of Rochester. I want to acknowledge and thank Ray Dorsey, who, as many of you know, has been a champion for digital tools and nervous system disorders for many, many years. And the PI of this study, Jamie Adams, who is at Rochester, and I'm very excited to represent this project, which started and has evolved over time thanks to feedback from the regulators. Very simple questions that were asked in this somewhat complicated study is to take patients very early in disease and ask if these digital technologies can be used to identify and differentiate Parkinson's from controls at very early stages, and ask if these smartphones and digital tools can help quantify both motor and non-motor progression in a 1-year duration of time. So very exciting, complicated study, very elegant in nature. Next slide. And won't have time today to go into the details of the study, but the two types of digital sensor platforms utilize the APDM Opal Mobility Lab. This has been utilized for many years in assessing gait and mobility across a range of movement disorders, as well as a custom developed iPhone app that partners with Apple Watch to assess both motor and non-motor symptoms in Parkinson's disease. The heroes here are Clinical Inc., who developed the platform, and the digital app was called Digital Artifacts, and Clario now oversees and leads the APDM system. Next slide. So the study itself was quite complex, but was very exciting in that as assessed in 1 year of about 100 patients were targeted, assessment of motor and non-motor progression across not just digital measures, but outcome measures, traditional measures, patient-reported outcomes, and even imaging biomarkers. What was unique for us at Critical Path Institute, our traditional approach to tackling data-driven solutions to drug development is to take data from either ongoing or legacy clinical trials. This was the first time we went to the regulators before the study even started. We had an incredibly engaging meetings. One was the Critical Path Innovation meeting with FDA. This was pre-COVID. It was standing room only with 9 divisions represented at a time where really we didn't have that cross-division collaboration that we're seeing today in the current framework. A similar forum took place at the Innovative Task Force meeting in Amsterdam. And what was so exciting in these limited 90-minute durations of time, we learned so much. And the FDA and EMA partnered together in giving us very concise feedback that helped shape this study in very new, innovative ways that was not in the original design. So I'll share with you just briefly some of the learnings we had from these meetings, and some of this has been published. Next slide. We heard so much around the technical issues, the challenges in transparency of algorithms. Some of the things we're learning just in this 2 days. This is incredibly difficult to do, and doing it on your own poses huge challenges. The agency made it very clear in this disease in Parkinson's very early, there are clearly opportunities to look at both motor and non-motor manifestations, and they were very keen in telling us to pursue those. Over and over, we heard the importance of linking and attending to the voice of patients with Parkinson's and how they are perceiving this type of technology and how it can help them. It's also, we learned very importantly that we needed to include normal controls. That was not in the plan originally because we didn't have budget, but the members of the consortia co-funded that. We're looking at normal control data. We just heard about that. And really exciting, we learned that in addition to the holy grail of an outcome measure, we really wanted to think about how these tools can be used to identify very early at-risk individuals. So the data is emerging. In fact, next slide is a snapshot of some of the data. This week, I think, should be the publication of the baseline findings from the WATCH-PD study. And this is just a snapshot of one of the particular symptom domains that we saw significantly reduced in Parkinson's versus controls at baseline. As a reminder, these people are quite early in the disease course. They're not on medications at baseline. And then halfway through the study, half of them will go on meds. So we have kind of a built-in positive control. Those data are being evaluated as we speak, but arm swing is one of the things that we saw quantifiable differences at baseline, as well as a number of other symptom domains that include finger tapping, tremors, speech, cognition. That study should be published, but these are just the baseline findings. And very excited that these results are now being analyzed to look at what changes over time. The next slide shows that we were asked and coached and advised from our regulatory agency partners, next slide, is to let the data tell the story, let the data tell the context of use. And it's so exciting that we were noted and found out after we saw the arm swing data that one of our esteemed collaborators, academic leaders, Anat Mirelman, had led a study carried out in people that are genetically predisposed to get Parkinson's disease, in this case, the LRRK2 gene. And in 2016 had published this paper showing that our arm swing can quantifiable, detectable differences in people with LRRK2 mutations at risk for developing Parkinson's. So here we have this example where the data tells the story, and we have 2 independent studies done over major long durations of time apart that are showing similar findings. Next slide. I think we're just almost out of time, so that I'll finish off with, I'm so excited that Jennifer Mayman will represent the work she's done with Jamie Adams and the team on how to link this project to the voice of the patient. And last is a call to action to tell everyone that we are not where we need to be in this field. Next slide. Just real quick, I'll share with you that there's an exponential increase in the use of these tools in trials. You've heard this from this meeting today. But to get to where we need to be, next slide, the call to action is to take what's been happening over the past in silos and individual organizations, work as global collaborations with the agency and with patients at the table. We work with people living with Parkinson's in the project. We need to agree on data standards. We need to share the data before and after the study, and we need to focus on the patients. So I think with that, I know I'm just about out of time, but maybe if you can pull up the acknowledgement slide, I'll just want to tell everyone that we're really excited to be in this point with this project. We're working carefully and excessively with more and more stakeholders. And I specifically want to call out some champions here listed: Jesse Cedarbaum, Bob Alexander, who started this project, and people living with Parkinson's, and Dr. Michelle Campbell, who's the catalyst for the entire project that you're going to hear more and more about from Jamie in a little, from Jennifer Mayman in just a few moments. Thanks, everyone.
Hi, Diana. You were exactly on time. I just popped on a little early. My apologies.
Oh, okay.
Great presentation. Really enjoyed it. Loved your message about engaging early with FDA. We also heard that yesterday. So I think that is really important. Fascinating to think about normative databases, and really makes a lot of sense with these new technologies. We don't really have an enormous amount of historical data of what kind of normal looks like the way we do with kind of more traditional measures or things that are used in clinical practice more regularly. So really interesting point. And of course, the patient centering is so important. So really appreciate it. And we're going to be talking more about this. And I know that there has been several questions yesterday, and I already see them in the chat today, about this device agnostic approach, trying to understand how we can make these easier to incorporate into clinical trials without having kind of one-off validation data needed all the time. So we'd love to hear a little bit about that too during the panel discussion part. Next we are going to be hearing from Dr. Abhinav Sharma, who is the Assistant Professor of Cardiology at McGill University. So Abhinav, I'm going to turn it over to you.
Awesome. Thank you so much for the opportunity to speak to all of you today. It's actually great timing. I was actually speaking with some of the Canadian counterparts and regulatory agency folks here, so it's great timing. So I really appreciate the opportunity to speak with you. My name is Abhinav Sharma. I'm an assistant professor of the Division of Cardiology at McGill University. Next slide. And today I'll be talking about the use of actigraphy in heart failure clinical trials. Next slide. Here are some disclosures. Next slide. So in terms of an agenda for today, I'll be talking about the role of actigraphy in heart failure, jumping to the evidence for some of the use of actigraphy measures in heart failure patients and in clinical trials, talk about the current gaps in actigraphy use, and a framework for how we can move forward. Next slide, please. So in terms of the role of actigraphy in heart failure, why is this actually important? Next slide. Oh, sorry, a little bit of formatting issue here, but the one critical issue is that the morbidity and mortality associated with heart failure is tremendous. In the orange line, this is epidemiologic data from Europe, but it's also been validated in the US and Canada, that if a person was hospitalized for heart failure, their 2-year mortality is approximately 50%. And if they were seen as an outpatient with heart failure, their 2-year mortality is approximately 25%. So the prognosis and mortality risk of these individuals is worse than most cancers. Next slide. And so with regards to digital innovation, the healthcare industry has been largely excluded until very recently from advances in digital health. The availability of powerful yet low-cost computing and funding from government and regulatory agencies and industries has resulted in major advances in the use of digital technologies. This helps to facilitate both clinical care delivery and optimization of clinical trials. Next slide. So this was a review we published several years ago, but still holds that the uses of digital technologies in trials has completely exponentially increased. And this trend holds true to today. So certainly there's a lot of interest in exploring the use of these digital health technologies in patients with heart failure. Next slide. And why is this actually important? Why actigraphy specifically in heart failure? Well, there's greater emphasis on the use of functional and symptomatic endpoints for heart failure clinical trials. This was a paper that I had the pleasure of working on that was led by the Heart Failure Collaboratory, which is an industry, academic, and regulatory sort of consortium that aims to advance the science in heart failure clinical trials. And of course, by including functional and symptomatic endpoints, this is much more patient-centric. It can increase the efficiency of trials, decrease cost, improve the data for statistical analyses, and we can get results before we can get hard morbidity and mortality data. Next slide, please. And another important thing in terms of, sorry for the formatting issue here, but why actigraphy is important? Well, as you can see in the blue slice, this is looking at where do people spend most of their time after being discharged from the hospital at day 30, 1 year and 2 years. And as you can see from the blue slice of the pie, going from around 40% to 70% and more, most of the time patients are at home or in a home environment. Yet we do a very poor job of characterizing and capturing what happens in the home environment. We're often focused on cardiovascular death, which is very important, and hospitalization, which is very important. But in fact, the time spent at home is actually where most people spend their time. They actually don't spend it in a healthcare facility. And Actigraphy provides an opportunity to understand what is happening in that period when they're not interacting with a healthcare environment. Next slide. And so going from the left to the right here, why Actigraphy can be utilized is, number one, it's much more broadly available. There's increasing comfort in the use of digital health technologies, the cost is going down, and there are companies that are developing devices for specific disease processes. The second is that current functional assessments, such as the 6-minute walk test, VO2, which has to be done with a cardiopulmonary exercise bike, has to be done in a facility with trained personnel. The ability to measure markers of actigraphy in patients' home provides an opportunity to democratize clinical trials. The third is that there's need for real-world data. We don't really understand what happens to people when they're in their home environment. And the fourth is that actigraphy can support the drug development cycle, not used as a way to substitute, but can provide additional information beyond hard morbidity and mortality outcomes, patient questionnaires, and let's say established functional endpoints. Next slide, please. So in terms of the current state of the evidence, again, with the Heart Failure Collaboratory, we published a systematic review looking at digital sensors for actigraphy in heart failure clinical trials. Next slide. Overall, we identified 11 randomized trials within our review. Next slide. And what we identified were three sort of major key outcomes. The first is that it's very unclear whether a lot of the devices that were used in these 11 trials were actually validated in heart failure patients. What that means is if a device says someone has traveled 3 kilometers in a day, how sure can we be that that's very accurate? And in fact, there was very little emphasis on validation of these devices before implementing them in trials. The second major issue was that there was extreme heterogeneity in the types of outcomes that were being measured by actigraphy. Some studies looked at peak movement, some looked at averages over a day or over a week, some looked at distances, some looked at step counts. So there's still a lot of heterogeneity, and there's not a clear direction in terms of a set of outcomes that are preferred for actigraphy. And the third is that there were actually some discrepancies. So for example, there were trials in which the functional endpoint was not improved by therapy, but the phase 3 and the phase 4 trial showed that there was a reduction in morbidity and mortality. And for instance, a lot of the trials did not accurately report how missing data was handled or how other actigraphy measures were handled. So certainly there's a lot of heterogeneity in the use of actigraphy in these trials. Next slide, please. So what are some of the future directions at this point in time, and how can we drive the science forward with regards to using actigraphy in heart failure studies? The first is I think we need some methodologic framework. Step one is to develop sort of a framework for validating these devices. And actually a lot of this work is already underway through groups such as the Heart Failure Collaboratory, which is an academic industry FDA collaboration, of which I'm thankful to be a part of, to develop frameworks in which we can actually provide guidance to both actigraphy device makers and other researchers for how to validate these devices. The second is what we really need to also understand is what is a minimal change that's acceptable for heart failure? What do I mean by that? Well, if someone is, let's say, getting a validated questionnaire, for example, the Kansas City Cardiomyopathy Questionnaire, which is a validated tool to assess patient symptoms and functional capacity for heart failure, or a cardiopulmonary exercise stress test, we know what change from time point A to time point B reflects a minimal change that we would consider clinically significant. But we don't actually know that for actigraphy. So the science in terms of creating a framework for understanding that minimal change criteria is quite important. And to anchor that to establish FDA recognized endpoints is also critical. And in fact, there was a recent call for applications to look at how we can include actigraphy in trials by the FDA. And I really applaud the innovation and the foresight for actually having these types of competitions. Next, I think we need to actually then take these methodologic frameworks and implement and embed them within studies. This doesn't need to be done on a lot of people, but actually cohorts of approximately 300 to 400 patients can provide a lot of insights into the use of actigraphy in clinical trials and to confirm and validate how the minimal change criteria frameworks that we have identified translate into other established functional endpoints, such as 6-minute walk test and VO2. And the fourth is evaluating the actigraphy measures against other FDA-accepted patient-reported outcome measures, such as I mentioned, the surveys like Kansas City Cardiomyopathy Questionnaire, and also explore the association of actigraphy with biomarkers as well. So these, I think, are where some of the future directions are headed. Next slide, please. Some studies that are underway that I want to highlight. At our institution, we're working with a Quebec-based company, as I'm in Montreal, a Quebec-based company called Hop Technology, and this has been funded by the Quebec Ministry of Economy and Innovation to understand how actigraphy measures that are measured throughout a day, over a 24-hour period, translates into a 6-minute walk test, and can we establish some benchmarks for how actigraphy can relate to a 6-minute walk test. Next slide. We also recently finished the Target HFDM trial. So again, sorry for the formatting here, which was a study I had the pleasure of getting involved in when I was at Duke University, in which we gave participants with diabetes and heart failure, so they had to have both, a watch, a Withings watch, and a mobile application that helped to nudge them to do physical activity. So this was a behavioral trial, but the outcome was all measured remotely by actigraphy. Individuals were randomized in a 1-to-1 manner, and the mobile health intervention was delivered over a 3-month period, and then there was 3 months of observation. Next slide. What our study identified as you can see in this chart here in the red line and the green line, was that individuals that were randomized to the active nudging strategy had significant improvements in physical activity as measured by step count. So our actigraphy measure was step count over 3 months that persisted. Individuals who were not randomized to it actually had a decrease in step count over time, and that was statistically significant. Very interestingly, their Kansas City Cardiomyopathy Questionnaire also decreased over time as well, suggesting that these individuals were functionally doing much better, or there was significant improvement in their Kansas City Cardiomyopathy Questionnaire, sorry. So what this goes to show is that we can actually successfully do actigraphy-based trials in heart failure. We just need to make sure that we're asking the right question in the right patient population. Next slide. So in terms of our conclusion, actigraphy can play a substantial role in advancing the clinical trials evaluating heart failure therapies. This has the potential to democratize trials, to improve the evaluation of real-world outcomes, and also supplement traditional clinical trial outcomes across different phases. Of course, there will be the need for collaboration between academia, industry, and regulatory agencies for more guidance and frameworks around actigraphy-based validation, minimal change, and association with accepted FDA-established endpoints. Next slide. Here are some of the acknowledgements in terms of our collaborations and some of the former institutions that I work with right now. And next slide. Thank you so much for your time.
Thank you so much. I really appreciate it. I love the fact that you kind of repeated the idea of what Jeremy talked about the importance of DHTs to measuring that feel and function idea. And the idea that movement can mean really different things. They can be very different measures when we say movement. I think that's really an interesting thing that I'm going to dive into it a little bit in the discussion as well. And then I loved your point about comparing against biomarkers. I think it was actually in a separate meeting a couple of weeks ago. I was hearing some discussion around, are there ways of more cheaply getting proxies for certain biomarkers? And I think that's a really interesting exploration way of thinking about it too. All right, and then our final presentation is Dr. Jennifer Mammon, who is an assistant professor of nursing at the University of Rhode Island. Jen, I will turn it over to you.
Great, thank you. So I'll be kind of picking up from Diane Stevenson and telling you a little bit more about the qualitative study that we did for Watch PD. Next slide, please. So we'll be looking at how do you assess the relevance of your digital health technology to monitoring symptoms that are actually meaningful to patients. Next slide. So this was a multi-stakeholder kind of effort. A lot of people involved in this academic industry, Critical Path, FDA, and this study was funded by the Critical Path Institute. Next slide, please. The rationale for the study is that we know that our digital technology are capturing fine variations in symptoms, but we really don't know if what it's capturing is actually meaningful to patients. And we need to be able to establish meaningfulness in order to get regulatory approval of these new devices for use in clinical trials. So this is pretty heavily noted in the new patient-focused drug development series. Next slide. The background for the study was the Watch PD digital study, which was a 12-month multi-center trial that Diane Stevenson's previously referenced, where they were essentially looking at the capability of smartwatch and smartphone technology to measure symptoms of early untreated Parkinson's remotely. We had both motor and cognitive function, and we'll be focusing on the actigraphy-based measures today. Next slide. So the two that we'll be looking at today are the walking and the balance test, and then the tremor task, which the methods for completing it are shown here. But like I said, we did have cognitive, speech, and fine motor tasks as well. These were smartphone and smartwatch-based measures. Next slide. So the aims of this study were first to identify what symptoms and impacts of Parkinson's were meaningful to people. So we wanted to come in with a wide open approach. And then our second aim was to identify whether the measures that we were using through these smartphone, smartwatch technology were actually relevant to measuring those important symptoms. And the QR codes here will allow you access to those preprint manuscripts. Next slide. Study design was a hybrid mixed method study. We basically took 40 participants. The total number of the original study was about 80. We took half of them and we enrolled them in interviews if they had exited within 6 months from the prior study. We started with a very brief survey on symptoms, followed it up with a one-to-one online Zoom interview lasting about 100 minutes, where we did symptom mapping, which we'll go into, followed it up with cognitive interviewing to assess the content validity of the tasks. And then we mapped little pictographs of the tasks back into the personal symptom map. Next slide. Demographics for our sample was predominantly white male, higher socioeconomic status with Parkinson's, approximately 2 years duration. Next slide. So this is what a symptom map starts out looking like. It's essentially a visual Likert scale. It's a hybrid data collection approach that allows you to collect qualitative interview data during an online interview and to categorize it inside of a quantitative framework. This allows you to effectively quantify the frequency or the prevalence of symptoms and how bothersome they are, as well as to associate what type of symptom your task actually relates to based on its association with a symptom. Next slide. So when we came into an online interview, the participant would essentially start by talking about all of their symptoms, which are represented in the heavily blue outlined boxes, and then they would delineate kind of what are the impacts of those symptoms. And we would work on one section at a time, kind of collapsing down sections in order to minimize information on the screen, but allowing us to capture in depth exactly what the impact of that symptom was. Next slide. The second step is once we had gotten all the symptoms in, and this included both spontaneously mentioned symptoms as well as things that we wanted to probe for that they may not have mentioned, we collapsed all the details down to just symptom nodes, and then we asked them to organize or to categorize their symptoms into this visual Likert scale. And we would have them move, we would actually do it for them, move these symptoms up or down and place them into different bothersomeness categories, as well as to rank order within the categories and to define what made things important to them. Next slide. We then followed up with a traditional cognitive interviewing assessment, which had 4 standardized questions. Did the participant understand how to complete the task? Did it relate to their symptoms? Was it similar to activities in daily life? And was it relevant to measuring the progression of their Parkinson's? Next slide. We then came back to the symptom map and we took little pictographs of the tasks that they had been completing, and we asked them to insert it into the map next to the symptom that they believed that it related to and captured, and to explain if it did not capture a symptom, what was going on and what was missed. And this allowed us to basically say, does this actually relate to your personal symptoms within the map? Next slide. Participants really liked this approach. They were very enthusiastic about it. We were able to give copies of the symptom maps back to people, which they very much appreciated. And they said this is actually a much more accurate and an easy way to represent their symptom experience, as well as to evaluate the relevance of the measures to them personally. Next slide. This is the coding structure that we used in order to analyze these maps. It's essentially associated with an underlying Likert scale. A patient-reported symptom score of 4 would be associated with most bothersome, and so on down. This allowed us to quantify both how often symptoms occurred as well as how bothersome they were on average. Next slide. This is the type of data that we were able to generate from these qualitative interviews. And you can see from this that activity-based symptoms actually were some of the most important in this early Parkinson's group, with tremor coming out on top, followed by fine motor, slow movement, and balance issues. Next slide. When we looked at whether or not the tasks were actually relevant to these important symptoms, we can see that tremor was relevant to bothersome symptoms for about 87% of people, but that walking and balance was actually only relevant to bothersome symptoms for 55% of people. So a little bit lower down, but still some of the highest. Next slide. On the other hand, if we looked at it in terms of symptoms that were important to people, some that they were actively wanting to monitor for, even if they were not currently aware of those symptoms being present, you can see that walking and balance actually climbed way high up on the list and became relevant to 92% of the people at their current PD stage. Next slide. So this led us to one of our key findings, was that symptoms can be important even before they're present. They can be past important symptoms or future important symptoms, and people may actually want to monitor for these. They may not be aware that they have them at this point in time. Next slide. So here's a classic participant quote, and they say, where people are saying they want to monitor important symptoms that they don't necessarily know they have. And they'll say, it's not so much what you have currently, it's about whether new symptoms develop, if they become more severe over time, did it change, they show up, or you didn't have it before, or maybe you didn't notice you had it. Next slide. The criteria for evaluating whether or not a digital measure was relevant was a little bit different than we think of from a trial standpoint, but they basically would say, it needed to be, did they understand what the measure was actually assessing? Did they think it was capturing a symptom that was important to them? And did they believe it was a good way to test those symptoms? And if you could check these three criteria, they would rate the task as being relevant. Next slide. So we're looking at the tremor task. This was relevant to 100% of people. One person says, I have tremor. I don't do the task this way in real life, but I believe it is a relevant task because it would pick it up. The other one says, I don't have tremor, but I understand why you're testing this, and I think it's important to test it. Yes, it's a relevant task. So both these people rated it as relevant. Next slide. For the walking and balance task, this was relevant to about 92% of people. And you can see here, this participant says, I don't have this symptom, but I'm worried about it. I'm actively monitoring myself for this symptom, so the task is important to me. So in this case, they weren't aware that they had it, but they were wanting to watch for it anyways. Next slide. So the take-home message from our study is really that transparency will increase the relevance of your digital measures. People, if they don't have enough information to assess, they will say that your task is not relevant. If they can't tell if it's picking up what's important to them, they will rate it lower on the relevance scale. Versus if they can say, I understand why you're testing this, and yes, it looks like a good test, it's going to become a relevant task to them. Next slide. So when you assess your task, not only look at content validity, but also think about the context validity from the patient standpoint. What symptom do they think it's measuring? Is it important to them? Does it limit them or get in the way of what they're trying to do? How bothersome is the limitation? Do they think it's a good way to test the symptom? Is it important to measuring the disease in general versus themselves personally, their progression at that stage in their disease? Next slide. Limitations of the study: higher socioeconomic status, predominantly white, and a single time point. So we'll need to replicate this in further, more diverse samples. Final slide. So basically, let's focus on what's important to people. Let's make it as transparent as we possibly can, and that is going to get us measures that are more relevant. Thank you.
Jen, thank you so much. Really loved this presentation. I know we had a lot of questions yesterday around how do you start to pull the patient voice in, and I love you kind of going through how you can do that in a more effective way. Really interesting to hear about what patients are concerned about, particularly that new loss idea. And would be fascinating to kind of understand, is it the things that they're most scared about losing it, or is it things where they feel like they could put safety measures in place, but they don't want to do that until they actually start to see that loss. And then and I think that this is actually makes sense, but I hadn't really thought about it. Some of these measures may feel really disconnected to what they're measuring and the importance of really communicating that. So yeah, I just really love this presentation. Thank you so much. At this time, I am going to invite all of our presenters to come back on camera, and we'll move to the moderated discussion portion of the session. So I I think we have everybody. We're waiting for Abhinav, I think. Okay, so, I think for the first question, what I would really like to hear is, we heard about how actigraphy can potentially improve upon existing measures for drug efficacy in some of these disease measures, and would love to hear particularly, what are some of the new measures that are available with DHTs, and what are you are most excited about? Which do you think are really kind of improvements over some of the more traditional standards that have previously been used? We had heard a lot about the 6-minute walk test, for example. I'm going to start with Jeremy on this one.
Sure. Thanks, Christina. And I think that, the answer to that question is that, overall, we're going to have a lot better visibility into what's happening in the life of the patient, even if the algorithm or the method that's being used is quite simple. And we're seeing a lot of success just with basic things like steps, but we've seen very advanced algorithms in our own use cases around macro measurements like sleep, the ability to get very precise on when someone is in bed and how long they slept and, when their wake times, and then more fine-tuned motor, measurements around tremor and dyskinesia and ability to see those things. We're getting much better with this technology in a controlled fashion to see these things. And overall, the benefit here is that it's just passive, right? I mean, we're not actively asking these patients to do any type of, active test in their home. Someone gave the example yesterday of coming into a sleep clinic and doing a PSG test and how that environment just completely changes your behavior, right? I mean, I wouldn't sleep good with all that on my head. And maybe I don't sleep good the first night if I have to wear a watch. And admittedly, that's a struggle for me. But after 2 or 3 nights, you get used to it and it becomes part of your routine. So just much better visibility in general around those things.
Would love to hear a little bit about about some of the nocturnal scratch work and what was the traditional measures that we've seen as well.
Yeah, I can answer that, or Steve can answer some of that. The nocturnal scratch work, we're seeing a lot of great validation happening around nocturnal scratch right now, and are able to see, as Stephen showed, even in some of his examples, the ability to see this very accurately, even with a noisy environment, right? You saw his example of things moving around, but not scratching, and our ability to detect that. Clinical validation is showing that we can get very accurate with that.
Steve, would love to hear your thoughts too. My internet may be going out a little bit.
Yeah, I mean, I think at the end of the day, I mean, it's about the patient, right? And I think that when we think about what kind of tool we're going to use, there's going to be so many instances where a risk-based sensor is going to be awesome, right? Because it does the trick. It's understandable. People are used to it. It's passive, and I think that for our perspective as technology developers, we think that, to put in the effort, it has to have value above something that is so approachable, like a wristwatch. I think in Scratch is an example where, finger-based scratching, specificity in daytime, we're seeing a lot of variation in night-to-night scratching. And I think that makes it more important to have accuracy and higher sensitivity. So I think it really is dependent on how you want to use it for. There's going to be a lot of instances where, even in the context of Scratch, a risk-based sensor will show you treatment effect with a drug. And maybe that's good enough. I think that it really comes down to what that fit for purpose is and what that ultimately, means for the patient. I think the point I want to make is that, I love the idea of how do we go back and make this useful to the patient who has to wear it? Wearables are useless in people that don't wear it. Same thing with drugs, right? If you don't take it, doesn't matter how good the drug is. And this is why I love seeing the behavioral intervention. This is why we're working on haptic, because as a researcher, as a clinical trialist, yes, we love the data. We're like geeks, like we love that stuff. But, I think ultimately it has to be about what is the way that we provide the most value to patients.
Jen, about any patient attitudes that you heard about around whether the DHT measures and the new more novel measures do feel more relevant, kind of going back to what you had said about what they make sense to the patients.
So that's a great question. They are relevant if they can tell what they're measuring. And if they can't tell what they're measuring, they're not going to say it's relevant because they say, I have no clue. So that's really the bottom line answer is if they can connect it, the more clearly, transparently connected it is to one of their important symptoms, the more relevant that measure becomes, which behooves us to be really transparent in terms of telling people what we're measuring and how it's going to be able to measure it. Because they'll sit down and dissect it and say, can this actually measure this? It doesn't look to me like it can measure it. And they get very skeptical.
Mhm. I think that
But-
makes,
Yeah.
go ahead, Jan.
Jennifer, one of the many great learnings from this experience in the Watch PD study is that the people that participated in the interviews learned a lot more about their disease on things that they thought may not even be linked to Parkinson's, and told us that they now can go back and help manage their disease in their daily lives in a much more effective way. But in Parkinson's, people get diagnosed relatively late and they have been reluctant to go into trials. So I think this, the ability to be able to recruit people and have them manage their disease and want to participate in trials to prevent the onset of many of these really frightening symptoms, including cognition and walking problems, is incredibly inspiring and going to help get more patients in these trials when we have more than 145 targets in the pipeline now.
Right. I'll tag on to that and just say the more that we're able to share data back to patients without compromising the trial, the more likely this is to become relevant to them. And that really is key, to treat them as partners rather than just someone, who's participating or contributing data.
Absolutely. I'm going to come back to, I did want to ask Abhinav, if you, particularly in the heart failure space, I know you've done several different trials in this, and I would love to hear your thoughts around, where is the difference in DHT kind of most important to you, that you're getting a different type of measurement, or that you're kind of getting that more continuous measurement and getting a better sense of what a patient looks like kind of on a more day-to-day basis?
That's a great question, and I think it's sort of a little bit of both. As I mentioned, the vast majority of patients don't spend the majority of their life in a hospital environment, right? 50 to 80% of their lives are spent at home, and so there have been many studies and even studies that we've sort of looked at, and when we talk with some of our patient partners, a lot of what they value is, am I fatigued? Can I do my daily activities? Am I able to enjoy the time that I have at home, understanding that I have a fairly progressive condition that could be managed, with appropriate therapy and lifestyle measures, but how can I improve the quality of life that I have as well? So I think Actigraphy can provide some clues into that element of quality of life and functional capacity and symptom burden as well. This provides additional supplementation of information beyond the traditional, let's say, morbidity, mortality outcomes when we're looking at those who've been hospitalized. Because by the time you're hospitalized, your symptoms have to be really bad, but there's a bit of a trajectory before you get there. It's not that you all of a sudden go from feeling totally fine to then not feeling fine, although that does happen. But in certain circumstances, there's a decline that happens over time as well. So these are all things that Actigraphy can give clues in because of that continuous nature of monitoring and addressing at people's homes.
Thanks. I'm going to switch gears a little bit because I see that there's a lot of comments in the Q&A box around validation and difference between validation and some of the choices that you have to make as researchers between commercial devices and kind of consumer-based devices and clinical trial kind of specific devices. And would love to hear about how you think, what are some of the components that you want to think about as you're choosing a device generally, and then also picking between sort of a more consumer-based device or versus a research-based device. Diane, I'd love to start with you. I know that y'all have done a lot of work on that the device agnostic space, and so we'll just start with you.
Right, thank you. This is a great timing for this question, and one of the reasons why we work across different industry sponsors, because they're all facing a challenge of how do they choose the right device. And I think the tech providers and the companies and manufacturers, we have Jeremy here, can really help us so much, because it doesn't make sense for each organization to try to figure out what is success looks like in terms of verification and validation. A lot of successes that have come out of EMA and FDA with the new guidance and framework, the DIME V3 framework. There's so much available information about what success looks like, but just true sharing of information that could help us align together and collaborate in what success looks like will help eliminate how to do this on your own. It's a lot of hard work, and we don't want to be guessing how many patients you would need to power a study to try to get that verification validation up front.
And love anybody else's thoughts on how do you, what are some of the steps you take when you're choosing a device? How do you think about what your particular patients might have be most interested in versus kind of what you're trying to get out from your study, for example?
Maybe I can
provide.
jump in.
Oh, sure. Yeah, I'll just give a quick point, because there's also several layers of validation, and really depends on what you mean when you say quote unquote validated. The typical suggestion of validation is whatever output you're getting, it is matched to some sort of gold standard, right? But there's different layers of validation that happens, in between. There's sort of an input validation where you can validate even each sensor, like is a heart rate, is the R to R interval in one device equivalent to some sort of gold standard, or does all that matter? What is the heart rate at the end of it, right? So I think we have to separate what component and what field are we validating. And the also critical question is, how much of an accuracy and what's the delta between the device and the gold standard that you would accept as being different, right? So if your watch says my heart rate is, I don't know, in the 80s, heart rate's a very important metric, and I'm using that, for heart failure, for instance. But let's say gold standard says it's 76. Is that validated enough, right? And so these are, I think, some of the bit of the unanswered questions, from the device technology point of view. Sorry to interrupt, please.
No, I think that totally makes sense. Steve?
Yeah.
I mean, I dog food our own products as a clinical trialist myself, doing a lot of sensor work on our own and not. And, I think the expectation should be high. Our perspective as a company is that, like, we should offer consumer technology level usability and acceptability, whether that's packaging to software, as well as our researchers, which are, I think, underserved in this context. But on the flip side, I mean, I think that the point is being made that, like, validation and proof is super important. So I think our perspective is, like, when you have a piece of hardware, for a lot of these use cases, I mean, they're medical devices, right? I think that it should follow the FDA's requirements at the CDRH level for safety and performance and, these kind of things, because it is, a physical medical device, whether that's the adhesive, the cytotoxicity, all these kind of things. And then when it comes to the algorithm, I mean, I think that's really important to validate in that context of use. Because heart rate is not just heart rate. Is it heart rate during ambulatory? Is it heart rate in cardiac patients? I think these are all really important details. I mean, for us, for instance, Scratch as an algorithm, whether it's a child or an adult, it's going to be different. Swallow, whether it's a Parkinson's patient or a healthy normal patient, those are going to be different. So I think it's really important to dig into those details and have high expectations when it comes to both the performance, acceptability, and clinical validation, because I think these tools have a lot riding on them, right? The drugs that we're going to develop, the drugs that are going to be used by our patients, on and on and on.
Yeah, the same rigor we see in other measurements. I mean, from imaging to ePro, there's a reason we use validated instruments and not SurveyMonkey, right? I mean, we can't bring new standards to what we do here because we're using technology that's available on the consumer side. And having participated in a Type C meeting, I can tell you the questions you're going to go all the way back to the sensor, and there better be traceability around each piece of that, how it was derived, how we can show inter and intra-device consistency among patients and their experience, and making sure that each device and each product and each outcome has a traceability around consistency. If we see Steve's performance, he did the exact same thing as me on a particular day, but he showed higher performance. Why is that happening? What's different? Does it have to do with where he's wearing the sensor, et cetera? So there needs to be, I agree, a high degree of requirement around this.
And that goes
And
back.
Christina, sorry, real quick example from the CPP side. So one of the many things the agencies both asked us to do is assess the source of variability in our study up front. So we have a paper published that came out on behalf of the consortium that really went through the Watch PD case example to say, what are the sources of variability? Because that's going to really contribute to that validation piece.
I think that part of that does come back to some of what Jeremy had talked about in his presentation, is that there's a couple, and Abhinav just brought up just now as well, which is that there are different components, and thinking about how can you do some of this validation in a less one-off type way. Would it make sense to separate out some of the calibration of the accelerometers within a certain technical specs versus the algorithm that then processes that into a measure? Or does it make more sense just to kind of keep that all combined when you're doing this validation?
I suppose it depends on what is the outcome you're hoping to validate, if I understand your question correctly. So if the outcome you're hoping to validate is an actigraphy based measure, whether step count or distance or what have you, that outcome can be validated. But if it's combining, let's say, multiple sensors together to create an algorithm that creates a novel biomarker, then I think each of those individual sensors needs its own validation before you can trust the results of that novel.
And I guess one of my questions is, we heard a lot yesterday about BYOD, kind of bring your own device, and the ability to use that. And have any of y'all worked on trials like that? And if not, what were some of the concerns you had around that?
Maybe I'll just highlight another Critical Path Institute consortium, that Electronic Clinical Outcome Assessments Consortium, that consists of all the different tech providers, and they come up with best practice recommendations that's going to help the field. And they've really tackled this whole topic of BYOD. What are the challenges? What are the limitations? How can we align and work together in this space? And I know in Parkinson's, there's a number of case examples that are taking place as we speak.
Oh, great. We'll be excited to see the results of that. Jen, I wanted to pull you back in there because I would love to hear if you have any thoughts around what patients have been saying around kind of having their own device versus being given a clinical trial device, and if there's preferences one way or the other.
So their preference is to use their own devices. So one of the things that people will say is they really object to having to wear two watches. One strapped to each arm. So that's very clunky and inconvenient for them. And there's just the kind of the fluency of, you're used to using your own device, you're comfortable with your own device. The more clunky it becomes to carry something else around with you, the less enthusiastic they tend to be about it. But they recognize they do recognize the need for that validity component and making sure it's the right device. So they're very aware of that. At the same time, they wish for the convenience of using their own.
And I'd love to kind of expand upon that, because I think one of the reasons why people like the idea of using their own devices is because they know it potentially gives them feedback. And I've been promising I would get back to this comment, because I'd really love to get some thoughts around what are some of the benefits and challenges in giving patients information about how are they doing on a day-to-day basis, but also potentially just returning those results at the end of the study, and getting that, have, even if you can't feel like you can't do it kind of at the beginning, is there a way of eventually returning those results back to the patient so they can see how what the results were? Jennifer, I'm going to come back to you to start, and then I'm going to ask the panel.
So I think returning at the end of the study would be really a fantastic first step. Even if you returned results to them before you did your final assessment of whether this was a relevant measure, would probably help for people to determine, oh, this actually was relevant to me. So I think that in and of itself would increase relevance. I think if there's an ability to return results during the study, that's certainly desirable, but people do recognize they can't always have that. So these are very intelligent people out there, and they know maybe they can't get the results during the study. On the other hand, I think as researchers, we need to be aware that they're already out there Googling everything. So they're talking to Dr. Google on a daily basis, and we think we're hiding information from them and blinding them to their participation. And what they're actually doing is turning into miniature researchers and going out discovering everything they possibly can about how this device might function and what their results could mean. So I think there's a little bit of naivete on our own part to think that we're actually truly fully blinding them. And we may be doing ourselves more harm than benefit in certain situations by withholding information.
Jeremy, would love to get your thoughts on this too. I'm going to go down the whole line though, just to give ample warning.
Thanks, Christina. Well, I mean, everything is about the patient, and just to be honest, I mean, Actigraph, in our early days of doing this, we made a lot of mistakes. I mean, the device was hideous, and people felt like they were in a research lab when they were in their homes, and so. Number one, there's got to be a lot of comfort there around the use of the technology and your comfort with it. But then also on the feedback topic here, what we found is that people want to feel like they're part of something bigger. And most participants in a clinical trial are in that trial, of course, because they want to get better and healthier themselves, but they also feel like they're part of something bigger. I mean, they've been sold that as part of informed consent. They've been sold that as part of the trial vision. And, you got that big logo up there, and they understand that what they're doing. So helping them feel like they're part of something and giving feedback around things like, hey, we got this much data from you today, and you contributed this much to the clinical trial in a meaningful way, doesn't impact behavior, but it does impact adherence and their ability to feel like they're engaged in something. So that's a very important piece. And then, lessons learned here is that we have found when we can give a patient profile, for example, at the end of a study, it's much more engaging for those patients and also for the site personnel, because the site personnel, don't forget, they're interacting with human beings. And we want to make them feel like, hey, I did my job. I gave somebody something today. It wasn't just take. And so there's a number of stakeholders that benefit from this sharing of data, and there's a right way and a wrong way to do it. If I start telling you got a steps goal today, then we're biasing your data. So we have to stay away from those things. And it goes back to the fit for purpose technology. I mean, I would love to just go buy off the shelf products and not have to make them, right? Hardware is hard, has that word in there for a reason, but it's got to be fit for purpose for those types of applications, if that makes sense.
Oh, totally makes sense. Steve, I'm going to throw to you. I'm particularly interested in that alert feature, that little motor vibration in your device that you mentioned.
Jeremy stole my thunder there. Hardware is hard, right? I think that, when we think about soft, flexible, wearable electronics, we think about how that can add more, right? So that it isn't another thing, because I'm, very cognizant in a position first that being a patient is difficult, right? It's hard, right? You have so many other things you have to worry about, another sticker, another watch, another whatever. And so it has to meet the value to the patient back. And that's why we're working on haptic and notifications and, therapeutic applications. Like one of the things we're working with the Michael J. Fox Foundation is to take that same idea and apply it to swallowing, to increase swallow frequency. And we did a lot of focus group work with PD patients because we were worried that they just were going to wear it. Like why add another thing? I'm pretty busy. But when you start unpacking the onion and thinking about, well, what really matters to you, which I love the previous talk around the things about eating and swallowing and speaking is so important to independence, that the feedback was resoundingly positive to say, I will wear this on my suprasternal notch in my upper chest because it will help me with an area that I'm really worried about. You know what I mean? So like there's that sort of risk benefit profile. And we all know that there's a high abandonment rate for wearable devices, right? For consumer fitness. I think everybody in this room, majority, has an uncharged Fitbit in their bathroom drawer. I think that's sort of like, a thing that we have to be cognizant of. So it really has to, match what the patient really wants and sees value.
Abhinav? I'm sorry, Abhinav, do you have any thoughts to add?
Oh, sorry about that. Yeah, I mean, I certainly would agree with all of those prior comments. The idea of fit for purpose, I think, is quite important. I think the BYOD approach can have a lot of merit, and it all depends on the specific question that is being asked. If the question is looking at real world experience and real world applications, a BYOD device could be quite important. If you're looking at maybe observational trends or natural history, that can be quite important. And I'm speaking from the heart failure lens. But if you're looking at, let's say, the results of a therapeutic drug intervention where it's randomizing between A and B or A and placebo or two drugs or two strategies, I think there may need to be greater confidence in the types of devices that are used and the results that are being generated from those devices. And so a potentially more fit for purpose device, or at least one that has gone through a bit more validation, may be more applicable. So I think all of these approaches have their own merits, and it really depends on the question that's being asked.
I think that makes a lot of sense. I'm seeing a couple of other kind of questions, but we are nearing our time. So I'm going to ask two questions and you can choose which one you want to answer. I see a lot of concern, and I think that we've seen this before too, about patients in these studies worried that if someone else picks up their device and starts playing around with it, how do is that data going to be recorded as them? I'm going to throw this to Jeremy to start with. Just, are there ways of differentiating who's wearing a device and that you understand when you get data that the person you think it is wearing it?
I get this question all the time, and it's a really interesting question. My favorite is, how can I tell my dog wore it and not, or if my dog wore it, not the person? Who are they trying to impress? Put it on the dog and get more activity, or I don't want to wear this, I give it to my kid, or I put it in the washing machine. So of the hundreds of trials that we've done, this is seldom an issue. And if it is, it's an outlier. But to answer the question directly, I mean, there are ways to discern data contribution, or I'm sorry, data attribution is what we refer to that as. And so we can see that happening with machine learning, artificial intelligence, detecting severe changes in the data or distinct changes in the data or gaps in the data. The bigger concern, though, is the biostatistics piece. So the worst thing that can happen to us is when we get a call and, hey, we want to put a wearable in a trial that starts next month. And nobody has talked about what is the statistical plan. Nobody's thought about what are some of the outliers, or what do we do? What's the plan around missing data or the covariates related to this data? This is complex data. This is not episodic data. A statistical plan is fundamental and super important at the beginning of a study in the protocol design phases. Otherwise, we're going to end up just doing a clinical trial with a wearable because somebody said it was cool. But if we plan ahead and we have statistical plans, then we can see what value this data is bringing and what type of measurements we're getting at, and whether or not too much missing data is there. What is too much missing data in a trial with a wearable continuous data capturing measurements 100 times a second, right? Somebody needs to define that. And that's why hardware is hard, Steve. So, I mean, there's certain ways to do that the right way and the wrong way. So I'll leave it at that.
I think that's great. And you actually got to my second question, which was really around how do you deal with missing data, and how do you think through, how much is too much missing data? Steve, your application was so different with Scratch. Are you also able to differentiate individuals?
No. I mean, I like Jeremy's answer, which is if you're worried about that, it's kind of like the train has left the station. It's never going to be the biggest problem. I mean, certainly we're looking at ways that you can video verify with our tablet. You're wearing it. The same thing with directly observed therapy for TB that's gone virtual now. So I think there's ways to solve that from a technology standpoint, but I don't think that even cracks the top 10 of biggest challenges when it comes to what we're trying to do here. That's my opinion.
Great,
All right,
Christina,
yep.
can I just comment here? I want to make a quote from my longtime friend and collaborator, Ken Merrick, leading the Fox Foundation's effort on staging now. And he says, the best way to avoid missing data is not to have missing data. So a lot of the things we talked about yesterday is to build that trust and relationship early on and have the patients engaged in understanding the protocol, the plan up front, and the issues that they're going to be asked to do to be part of the trial. It's a partnership with patients from the very beginning. And I think so many of these questions and gaps happened because of the approach that Jeremy said. People just put it in and saw what happened. And I think we're in a whole different world now, thanks to this engagement of the patients up front.
Oh, great. I really appreciate it. And Jen, I'm going to give you the last word. I'd really like just to hear from you, kind of your last thought on what you really wish everyone would do to help center those patients.
Well, that's a big ask.
And in like 3 words or less.
3 words or less. I would say it comes back to what Diane just said, patient engagement. We need to know what, if we're doing this to benefit patients, we need to know what they want. We need to have them intimately engaged throughout the process, and we need to treat them as partners and maximize our reciprocity as much as possible, because the more engaged that they are, the more likely they are to partner with us in the collection of accurate data. So people will engage in fewer cheating behaviors if they know why they shouldn't engage in cheating behaviors. They will wear their devices more consistently if they know why it's important to wear it, particularly if they know what it's measuring. So all of those things, really just treating this as a partnership rather than us conducting research on people, I think would be key.
I love that answer. And I think having that statistical plan that assumes that people are people and aren't going to be perfect as well. I think kind of the combination of the two, I think, is really where we want to be. I just want to thank this panel. You all have been fabulous. We've got a couple minutes over, but we are going to continue to have our break. It's going to be probably about 5 minutes now. So we will be restarting at 3:40. And thank you again to our presenters. Hi, everyone. I am going to apologize again. We are having technical difficulties. And so you may have heard from friends and families and colleagues who may be trying to join the Zoom who may be having difficulty. But we do want to make sure you're not just sitting in silence. So we are going to bring Jennifer Goldsack on the line. She is the CEO of Dime. And what's supposed to be the session 4 moderator. And we're going to have just a quick chat to somewhat extend the session 3 discussion that we did, and also potentially talk a little bit about uses outside of the actigraphy space. Jen, thank you so much for bearing with us during these technical problems. I have heard we have several major IT departments working on this issue, and hopefully we'll be able to get back to regularly scheduled programming. But Jen, welcome so much. Thank you so much.
Oh my gosh, of course. And there's nothing quite like being the tech enthusiast on the line in the middle of a tech disaster. So yes, happy to be here.
So I think one of the things that we would love to hear about is, where are some of the most promising areas that that you see for the future use of sensor-based DHTs? And maybe actually just start us off by talking to us a little bit more about DIME. I know we talked about it yesterday. I know that Lucy talked a little bit about yesterday too, just for people who weren't here yesterday.
Yeah, always happy to talk about DIME, Christina. So thank you. So for folks who don't know us, the Digital Medicine Society, or DIME, is a global nonprofit. Our mission is to advance the ethical, effective, equitable, and safe use of digital technologies to redefine healthcare and improve lives. And Christina, I think the most important thing about our organization, and it extends to this conversation in particular, is that we're not tech determinists. Our goal here is not to shoehorn digital solutions into every nook and cranny of healthcare delivery and clinical trials, but rather what we think about are some of the pressing and persistent challenges that we've struggled with across the clinical trials enterprise, across healthcare delivery. And that's where we focus our efforts. I intentionally continue to mention healthcare delivery in the context of this conversation, even though I know we're talking about DHTs in clinical trials, because the difference between the digital era and the, analog, if you will, the pen and paper era of healthcare is that, I remember the IRB applications and, your clinical data would be in a manila folder, locked in a filing cabinet drawer, and only the PI would have the key. And then, of course, all of the statistical analysis would happen on a non-networked computer. And that's simply not the era that we live in now. And one of the fantastic things is that data can flow. With the appropriate permissions, we have the ability not just to capture data outside of the clinic, but to extend the way that we use every data set, again, with the appropriate permissions. So we've been thinking a lot about, the use of real world data and evidence for a long time, but there's a moment here where we stop thinking about the use of data in clinical trials in one silo and the use of data that we use to improve the way we care for people separately. We have to be thinking about these things together. And when I reflect on the tremendous remarks of the last panel, the focus on the patient, the participant, the individual, the usability, the reliability and quality of the data, those are characteristics that will serve us well across the clinical trials continuum. And we've got a real opportunity here to think about measuring health differently through DHTs.
And I know we talked a lot about accelerometry and actigraphy in the last session. And I wonder if you could talk to us a little bit about some of the other examples that we see DHTs being used in.
Yeah, and so, there's some lovely presentations hopefully coming up that include things like continuous glucose monitoring, that include a variety of different sort of sensors that might be available on sort of patches. We can think about, our ability to do cuffless blood pressure monitoring. We can think about our ability to do, or quite frankly, in some cases, not do things like pulse oximetry using these sorts of sensors and tools. And then what's really interesting, and Dina will present on this, and I think this is where the really exciting sort of future lies, is those non-wearable technologies, right? How are we thinking about those ambient sensors? We're quite poor sometimes at the language we use around compliance and adherence, and we're a little bit patient and participant blaming. And, again, on the last call, people were talking about that uncharged wearable that someone has in a drawer somewhere. I mean, the ideal solution is that actually we reduce all of that burden and we start to depend more on these ambient sensors that actually don't require a conversation about compliance. You sort of plug them in and you leave them. Now that raises all sorts of privacy concerns, but let's talk about the bed mats. Let's talk about the smart scales. I mean, for heaven's sakes, there's a smart toilet out there now. All of these things already exist. And I'll give you an example of where this has been really powerful, Christina, and hopefully that will be some fodder for conversation. So at DIME, we have a tremendous, nearly 3-year-old partnership with our colleagues at the VA. And the VA have really been focused on reducing diabetic foot amputations. And think about the context of this. Think about the fact that the VA is responsible for individuals who have come home from the theater of war and they are now retired war fighters. So imagine coming back and you didn't lose a limb in battle, but because we failed to care for you appropriately and your diabetes, you actually end up losing a limb because of the healthcare system. They have a really fantastic piece of technology. It's a smart mat, and it measures the temperature in your feet. And every veteran with diabetes now has access to one of these mats. They typically put them right by the sink. So in the morning or the evening when they're brushing their teeth, they're able to get a temperature read on their feet. It's able to trigger outreach if we see any deterioration in temperature, which is a proxy for circulation. And Christina, the extraordinary results of this work is not a single veteran who's had access to one of these mats has lost a foot.
Wow.
We have absolutely eliminated the need for amputations as a result of these technologies. So this is the promise. This is the power of these tools. They're not acting alone, right? We need the individual to interact with them, but the more simple we can make it, like stand on it as you brush your teeth, put it somewhere where you can't help but do it on a daily basis. But it also requires us to deeply understand what the data means. In this case, what's the threshold for risk as the temperature starts to change? When do you reach out? And, what do we do in these situations? And so that's an enormous success story, and we have to be able to draw on that. But for me, what I'm particularly excited about are the new tools and technologies, the second generation, if you like, that are much, much more patient-centric, whether it's, Jeremy, applaud the work that Actigraphy has done, sort of put his hand up and said, when we started, it wasn't good enough for patients and participants, but their latest watch is absolutely fantastic. The battery can go to 30 days. The appearance, the user experience is absolutely world class. So if you have to wear it, make a tool like that. And if there's a way that you can avoid having to wear it, then that's always optimal.
I think that makes a lot of sense, and I would love to hear. I know that y'all have done several toolkits and frameworks and things like that around some of the validation issues that we talked about, and this is something that came up at the end of the session too. Kind of having a statistical plan for that missing data, particularly in a situation where we're not quite at that space where it's more passive, and so you have to have more realistic expectations of people are going to forget to occasionally to charge their watch. I mean, that's just going to happen. People are going to leave home without it. And that's just going to happen. And so how do you think about it as you're designing some of these plans? How do you think about what some of the allowable, what some of the allowances might be around missing data? And if you've had, if you know of any experiences kind of with what FDA has been thinking about this, would love to hear about that as well.
Yeah, and I think my colleagues in the last session did a really nice job of bringing up some of the key issues, which is, all too often we don't allocate enough time to do the thoughtful, intentional sort of statistical planning that's necessary in order to be able to handle the data quality issues. And, one of the things, and Christina, you've heard me at the table on this before, so forgive me, I'm just going to do it again, which is the quality of data we currently rely upon isn't perfect either. There are missed clinic visits. We know that there are challenges around self-report, the timeliness of that data, the completeness of that data. This is not a new challenge, and it's certainly not unique to technology. But what's interesting about technology is with the appropriate time, we can ameliorate almost all of these challenges. So, and I'm going to be a brat, I don't want to hear what happens if we put it on the dog again. I mean, if your algorithm is so bad that you can't distinguish between a dog's gait and a person's gait, then you really shouldn't be using it in a clinical trial. So there's all of these, and think about it. We only need 90 seconds of your gait data to be able to personally identify someone. Now, that's terrifying from a privacy point of view, but it's very good from the point of view of data attribution, because even if you aren't using, gait data and you're interested in something else from a smartwatch or a patch, you can use that as your sort of gateway to data attribution. So that's more than possible. And then when we think about missing data, there's been some really terrific studies, and I always reference a study from Aidan Doherty from the UK Biobank, who actually did some really great work to define what do we even mean by missing data. This data is so rich, it's such high resolution, right? If, for example, we are interested in gait speed or characteristics of gait, do we actually need more than 1 or 2 intentional bouts of walking every single day to be able to answer the clinical question that we're interested in? So first of all, we actually have to define what we mean by missing data. So the data quality issues, I think, are largely solvable, but you have to allow yourself time. There's going to be a lot of time savings downstream when we use these DHTs in clinical trials. We're going to be able to recruit more quickly because they're lower burden. We may need a smaller sample size only because the data is so much more rich. It's such higher resolution. But we are going to need to spend just a little bit more time up front to position ourselves for success and make sure that we don't end up with very expensive data that we simply can't use.
And actually, I'd love to dive a little bit into that smaller sample size. I think that came up in the last session too, and would love to hear a little bit about why would you potentially have a smaller sample size in this case? I think that everyone loves a smaller clinical trial in terms of quickness and efficiency.
There's some very senior people within FDA who, and I'm being silly, who love a big trial, and we all love a big trial because the more
efficient.
exactly. But the reality is, the clinical trials enterprise is struggling, right? We need to figure out how to reduce the cost of clinical trials and bring therapies that actually work and address the symptoms that matter most to patients to market as quickly and affordably as possible. And we need to make sure that the data with which we're making decisions about whether to bring a drug to market in the first place and then whether to prescribe it, that information is generalizable to every single person that our field exists to serve. So why is it so exciting? So I would say that it comes from two issues, and frankly, both are related to sort of the resolution of the data. So the resolution of the data doesn't require that we wait that there's a 16 visit schedule and we are sort of limited to these snapshots of data. And that we have to sort of string that out over a longer period of time. We can think about what's the mechanism of action of this medical product? When do we expect it to start taking effect? And we can directly measure on that timeline instead of a study sort of visit timeline when we expect to see a change. The other piece is with this higher resolution data, we're going to see things that we haven't seen before. So we are going to have a clearer signal, which, a bigger delta, which is always going to allow you to reduce that sample size. So there's sort of two things at play here. And there's a really tremendous paper, actually, and it's got a great title, which is why I always remember it. It's called Moneyball. And it actually takes examples in Parkinson's and the use of accelerometry in Parkinson's. And it actually walks through a sort of statistical analysis with a variety of different assumptions. And it sort of assumes that we're going to be able to identify more correctly with these higher resolution measures when we are seeing a signal in the data, when we are seeing a therapeutic effect. And that's what's driving primarily the potential reduction in sample size.
And I think some of what we've been interested in thinking about DHTs for, I mean, oftentimes when we talk about it as endpoints. But I think one of the things that got brought up yesterday and potentially could kind of go to that smaller sample size too, is patient selection and being able to be more precisely kind of measure, find the people who are going to most benefit from a particular new drug. And would love to hear about any examples or any thoughts you have in that space too.
I mean, absolutely. And I'm so glad you've brought that up. And, quite frankly, while we need to be sort of evidence and science first, and that's obviously the purpose of this conversation, we have to make sure that these things are sort of economically viable. And, I'm of the opinion that if all we're doing, and I say that in inverted, in sort of quote marks, because it's still an enormous lift, but is relying on these measures in order to give us a signal about the safety and efficacy of a product, we simply aren't going to see the ROI that we need to in order to continue to warrant the ongoing investment that's necessary in developing the evidence, in developing these new products, and being confident that we can trust them and that the data that they read out. So to your point, we have to be thinking about, when we're deploying these tools and we start to see signals in the data that perhaps there's a group of, responders in that data, that can start to identify, a digital phenotype. And I think about a diagnosis like major depressive disorder, for example. 1 in 5 Americans during their adult lifetime will get a diagnosis of major depressive disorder. Some people respond very well to medication therapies, some don't. major depressive disorder, I would argue Parkinson's, I would argue, Alzheimer's. That's not just one disease, that there are multiple sort of sub-diagnostic categories in there. And if we can start to tease them out, then what we can do is feed that information back upstream in the development process. So we can start actually being more targeted in our hypotheses. Just as you said, we can then start to use these digital measures as sort of screening and enrichment tools. And then we start to see, and this gets back to a potential smaller sample size, right? If you're bringing the right patients in who are most likely to respond to a new molecule, you're going to see a much stronger signal in the data. You're going to be able to reduce your sample size. And then if we really want to get punchy, and that's my favorite thing, we can pull those measures into the post-market, right? So we can start to use those as screening tools during routine care. And maybe even we start to use them to do things like, value-based coverage. So there's all sorts of value and benefit that can return to patients, that can reduce timelines, that can make, what do we have, like a 5% rate of technical success in drug development right now? I mean, there's so much promise here. As I said, these things aren't a silver bullet that are going to solve everything, but there's enormous promise. And we do have to think about the entire clinical trials development life cycle.
I think those are all.
I did want to touch on one other question from the last panel that I thought was really interesting. So there are a lot of questions around, are there some ways that we can do some of the verification steps? So in particular, evaluating the underlying hardware and the performance of particular tools in a way that I would describe as almost sort of commoditizing that step. Can we a priori say, here's a list of, tools that would work very well with this algorithm, for example? And I feel pretty strongly about this. We only need to look at what our colleagues in CDRH have done in the way that they've, very, very effectively, I think we'd all argue, the way that we look at software, SAMDs, so software as a medical device. By definition, that's simply a piece of software, but software can't exist as an inanimate object. And the way that they handle that is to say, either there is a set of technical specifications that any underlying piece of hardware has to meet in order to be used with that SAMD, or there is a whitelist of products that's maintained. And I think that is proven successful. And there's absolutely no reason why we can't translate that approach onto the drug development side of the house, that we can't use that to power both BYOD approaches and think about, is there a way that we can start to commoditize some of the underlying hardware? When we built out the V3 framework at DIME and with many colleagues from across the industry, including some folks who have already spoken, what we really wanted to be able to do was to sort of make the tech stack modular, that we wanted to be able to interchange a technology that was fit for purpose for the very particular patient or participant population that we might want to serve, recognizing that different people with the same diagnosis may prefer different products. But as long as the data quality is the same and replicable across different tools, we should be able to substitute those things out. And I think Jennifer made a very, very sort of compelling argument that we need to meet patients where they are with these sorts of things, that we should then be able to pair those with an algorithm, and we should be able to do clinical interpretation of this data, in one patient group, and then potentially translate that into others without having to go back down the whole tech stack.
I think that makes a lot of sense. And I think that there is a lot of interest in trying to see, can we shorten some of this verification process around DHTs? Can we make it so that it's more straightforward to include them? And I think, we heard some of the differences between traditional devices and sort of what you're expected, what kind of the expectations are around traditional health system devices, verifications versus DHTs. And I think, probably part of that is just because these are newer, right? And there are kind of more, there are higher standards around calibration and testing for some of these hospital-based systems. But I'd love to hear your thoughts on kind of what some of the similarities and what some of the differences might be. Yeah,
and I think that, I find myself often torn between, do we want to slow down the pace of adoption by saying it has to be better than it is today? On the one hand, I think there's such promise in these tools in terms of how they can drive easier access, for example, to trials, how they can reduce costs and timelines, as we've been speaking about, that if it's a net, maybe that's an okay place to start. And then I have a change of heart 5 seconds later and, feel like we need to do a much better job. But I think that, the key to unlocking this success is being very intentional. I think it's absolutely critical that we don't create a two-tier system where there's the tools that are developed and deployed for healthcare delivery and for caring for people during routine clinical care, and the tools that we rely upon for clinical trials. Now, that's not to say that we should lower our standards for the data quality and trustworthiness that we rely upon in clinical trials. But if we want to create a future where we aren't asking people with cancer, kids with rare diseases, to come to the clinic on a Tuesday to participate in a clinical trial, because that's one set of data. And then, for the kids, we pull them out of school again on a Thursday because they have to see their doctor on the other side of campus. If we want that data to flow, if we want to be able to reach people wherever they are, if we really want to start doing pragmatic research and use, what you guys, frankly, are the leaders in the field on, sort of talking about real world data and evidence, if we want to include sensor generated data in that, we absolutely cannot do it with a siloed approach. We have to be thinking across the board. And what's fantastic watching our FDA colleagues lead over the last few years is the success for the Digital Health Center of Excellence, and how that sort of steering committee, which is mentioned and championed in the recent framework that was released, talks about having this steering committee of folks from across the agency who can come together, and we are thinking in the same breath, what does good look like when we're using digital drug development tools, and what does good look like when we're creating diagnostic and monitoring tools to care for people even outside of the clinic.
I mean, I think that what you said makes so much sense, and I think exactly what you're saying. I do see a lot of the similarities between real world data, kind of more writ large, and a lot of these DHTs, whether be in both cases, and that not necessarily wanting to have kind of that siloed approach. I think that all makes a lot of sense. I do want to shift gears just a tiny bit and ask you about special populations, and where DHTs, and we talked a little, and this was brought up a little bit yesterday too, about giving people without a voice a way of being able to give that information, through measurements to a health system, as opposed to kind of relying on someone else reporting on their symptoms. And would love to hear, any thoughts you have about kind of those populations and some of the benefits there.
Yeah, and I mean, I think this is so important. And and actually our team has done some really tremendous work that I'm very proud of. And, I think this starts at the very beginning, as Mary Poppins would say, a very good place to start. So, when we start thinking about giving a voice to these special populations, or in fact, giving a voice to anyone who healthcare delivery and medical product development exists to serve and care for, we have to take an inclusive approach to developing these products. We have to think about the problem we're trying to solve for and for whom. And we need to make sure that at every single step of developing these new DHTs, we are hearing from and being driven by every single potential user of these tools within a particular context. So that should absolutely be table stakes, but it's not enough to have a product that you can take off a shelf, or perhaps it's a selection of products, right? We were just talking about, can we switch these things up? The nice thing about digital is the ability to do hyper-personalization, right? We give people exactly what they need, as long as it doesn't affect the reliability or the consistency of the data that we're gathering. We're just giving them every opportunity to participate in a way that works for them, the limitations of their sort of health condition in their lives. So, the development of these tools is really important, but it's also the deployment of these things, right? So there's some fantastic science coming out of, and I'm now actually answering one of your earlier questions about what are some good sensors coming down. I think some of the vocal biomarkers, the voice biomarkers, are very exciting indeed. We also, through the course of the pandemic, we saw so much around cough counting, for example, and cough characterization. You can think about that from, diagnosing or monitoring infectious disease, or maybe getting an early read on something like lung cancer. Still the number one cancer cause of death, unfortunately. Absolutely extraordinary. So, that's great. And we could even give you a device. We could be very clever and make sure that everyone has one. But if you work on a building site where it's really loud, that's not going to work for you as well. If you work in an environment like my husband does, where you can't bring a device into the office with you, right? That's not going to work for you. So, we need to sort of build intentionally by design. We need to make sure that we aren't excluding anyone. But I genuinely believe that these are solvable problems. And I also think that, And we can talk about the sort of, challenges of building tools that work for everyone, or we can talk about an opportunity to say, we don't have terribly representative trials right now. We're not terribly good at reaching people. We're not terribly good at giving a voice or generating data from and making decisions that actually contemplate enormous parts of the population. We're at a watershed moment. We've got, some of the best and the brightest, both within the FDA and now in industry, in academia, working on this sort of next generation of tools that can power a different way of evidence generation. Let's just insist that we bring everyone with us. Let's do it through good product design, and let's do it by, developing protocols in partnership with communities. By the way, please pay them when you ask them for help. And then, let's do this right. It's such an exciting moment, and we really can shine a light on, learn from, and serve populations that we've simply ignored because it's been all too easy to date.
Jen, that was a great answer and a perfect timing, because I believe we have been able to pull our panelists from session 4 into the session. It looks like by telephone. So we are gonna go ahead and try and go back to move back to session 4, which Jennifer, you're moderating. And so I hope you might be able to just, I'm gonna go off. If you can just go ahead and introduce the session and then introduce the individual presenters to do their presentations. That would be great.
Absolutely. I'd be happy to. Christina, this was fun. Let's dive back in with the agenda. So in this particular, I'll spare everyone an introduction. I think you've already heard quite enough from me already. But what we're going to do in this session is we're going to talk about different types of sensors that are being used to measure outcomes across different populations. And in particular, we're going to talk about sensors beyond the accelerometer. In this particular session, when we talk about diversity and accessibility, we mean it in the broadest sense of the term. So we're thinking about age, gender, socioeconomic status, race, ethnicity, and other socio-demographic features as an example. And let's not delay further. Our first presentation will be from Dr. Kuldeep Singh Rajput, who's the CEO of Bioformis. So Kuldeep, I'll turn it over to you and looking forward to your presentation.
Hi, everybody. This is Kuldeep here from Bioformis. Thank you for the opportunity. So, I'm not able to see the screen, but I will, flip through the slides. So, first of all, in recent years, we have seen the level of innovation and growth in wearable technology has led increasingly to the use of wearables and other digital health tools and in clinical trial setting, as well as in healthcare setting. So if we go to my next slide, over 1,500 clinical trials have been conducted since as of 2020, where a wearable biosensor has been utilized, and I foresee that this number will continue to grow as trials are conducted virtually and the data is gathered remotely. So if we go to the next slide, as wearable biosensors and DHTs continue to prove their value in various care delivery settings, either in the hospital or in the home, we see significant promise and opportunity in drug development and clinical trials. Collecting this continuous, physiology data from participants using sensors in real-world setting, which is often not collected, may fundamentally change how clinical trials are designed and conducted. For example, in early clinical drug development, collecting continuous data may enable us to identify early safety issues, inform drug adjustment or dosing, and sometimes even lead to discontinuing some of the drug candidates, which is significantly valuable to biopharma partners. So on this slide, you see various applications where you have, safety and patient phenotyping is one of the applications where sensors could be utilized. Novel endpoints, for example, things like mobility and actigraphy, which we, already spoke about, sleep studies, various surrogate endpoints associated with cardiovascular, could be, used. We have medication monitoring and intervention, things like improving adherence, dosing information. And finally, the biggest value of wearable biosensors is really how do we drive patient enrollment and engagement and retention in a clinical trial when trials are done outside the four walls. So if we go to the next slide, I wanted to highlight our framework, how we look at building a platform or a technology platform that supports clinical trials. The platform basically has two major components. The first one is the technology elements to it, where you have collecting data from the patients using sensors, companion app, and an ability to collect physiology data from patients either continuously or episodically from various FDA-cleared wearable biosensors. The second module there is what do we then do with the data? And it was extremely important for us as we thought about trials and remote data collection to put a framework in place which has multiple elements to it, starting from what raw biosensors are we collecting? For example, electrocardiogram, photoplethysmography, accelerometer data, as well as other vital signs data. Then once we capture all of those, how do we derive certain novel digital endpoints which could be potentially used in a trial? Most of the trials, and the sensors which we focus on are multivariates. So when you capture all these continuous data, how do you look at multiple physiological signals and start looking at changes in baseline, at the trial initiation as well as the completion of once the monitoring is complete? How could we use some of the wearable data to either detect or predict a clinical event? And once that event is detected, what do you do next? How those alerts are managed and how the interventions are provided. Of course, all of the information, what is analyzed eventually, is going to be part of the data lake with the right integrations into EDC platform, CTMS platform, and of course, a dashboard for investigators and study coordinators. So that's the first framework from a platform point of view. And the second key aspect we all need to be thinking about is how do we operationalize this? Things like how are kits being and devices being sent to patients' home? How do we do the logistics? Do we have a virtual command center which focuses on managing compliance of the patient, tracking safety, because we are capturing enormous amount of continuous data, and how are we tracking and managing safety of these patients? And are the clinical personnel available to be able to do that? And finally, of course, delivering certain in-home services in virtual trials if needed. So if we go to the next slide, another piece of information and framework, which has been increasingly important is sensors are just going to proliferate. Wearables, we have seen a number of sensors over the past 10 years, sensors which capture single parameter, and now we have increasingly started to see sensors that have ability to capture multiple physiological signals using a single device. But it's extremely important, to decide which sensor is the right sensor for your clinical trial or a clinical study, depending on the duration of monitoring, the patient population, and the parameters interested. So typically, when we look at a study protocol, and investigators and pharma partners look at study protocols, there's a set of specific criteria we have come up with, which involves, okay, what raw sensor or what raw signals the device captures, what's the form factor, what is the connectivity protocol, either it is, Bluetooth, cellular, is the data being stored, on the device, or the data is being continuously transmitted, what is the data quality, is it regulatory cleared, what's the battery life, etc. So as we see proliferation of sensors, having a framework that enables us to decide the right wearable and the right sensor for a study becomes extremely critical. So what I'm going to do now is give you 3 or 4 examples of some of the trials where sensors have specifically been, utilized in studies. If we go to the next slide, I'll start by focusing on cardiovascular trials, specifically where, endpoints are used primarily for safety monitoring as well as some surrogate endpoints. And this is a quick overview of what you see on the screen, all the places where digital technologies have been used in cardiovascular medicine or cardiovascular research. For example, arrhythmias, arrhythmia detection, things like, QT, QTC, and so on. If we go to the next slide, specifically what I have done in this slide is taken a very similar approach of data being collected from the sensor and how do we stack multiple capabilities on analyzing the data for two example trials. The first trial, what you see, is a trial evaluating safety and efficacy of an infusion nitroxyl in hospitalized patients with heart failure. The primary endpoint of that trial was clinically relevant hypertension, and the secondary endpoint, specifically was a combination of lab readings as well as physiology-based measurements. And if a digital health tool would have been utilized in a trial, the way we would really do it and the capabilities would be, involving a patch-based sensor. For example, what you see on the screen, a vital patch, which has an ability to, collect things like electrocardiogram, heart rate, respiration rate, temperature using a single device, as well as an episodic blood pressure, monitor. Once those data is captured, we are then able to utilize those, continuous signals to derive shortness of breath, dyspnea, hypotension, physical function using accelerometer data. One of the secondary endpoints in the trial was change in vital signs from the baseline. So, using FDA-cleared software as a medical device called BioVitals Analytics Engine, you would be able to look at changes in physiology, for example, heart rate, respiration rate, activity, from the baseline in patients. And finally, as a safety endpoint, ECG morphologies were, included, for example, QTC, QT, PR segments, QRS durations, etc. So here's an example of how do you take, a wearable biosensor and make it, and put it in the context of a clinical protocol or a clinical study where you're capturing the data, but you're, also trying to utilize that data for both safety and efficacy, of a trial. And the second example is, again, safety and efficacy in another drug trial where, the evaluation was focused on, paroxysmal SVT conversion to sinus rhythm after an intervention. In this case, you would either use a continuous, a patch-based device to capture EKG or an episodic, device pre and post therapy, where you would detect various different types of cardiac arrhythmias, in this case, SVT, as well as sinus rhythm, which would provide an ability to understand the efficacy of a trial. If we go to the next slide, I wanted to give you two other examples, which are not related to cardiovascular, but one example specifically, again, focusing on safety monitoring in oncology. We have seen significant growth in T-cell therapies, and here's an example where a digital health tool, is being utilized, or digital health tools are being utilized to evaluate if a T-cell therapy or a CAR T can be, administered and managed in an outpatient setting. In this example, or in this specific study, it is really a feasibility to treat patients with Yescarta, in outpatient setting. Sensors are being utilized to collect continuous data, which involves blood pressure, heart rate, respiration rate, temperature, using the data, evaluating, different kinds of models. Are we able to detect CRS events or cytokine syndrome response events early enough so that T-cell therapies and patient safety is managed appropriately in an outpatient setting so that appropriate actions can be taken. The second example I wanted to provide, if we go to the next slide, was another example of a digital health tool as a novel endpoint. And as we all know, pain is measured very subjectively today using NRS scale, numeric rating scale, which is 0 to 10. However, those are very subjective, have various biases, and can vary over time. And in this specific study, in specifically patients with endometriosis, a novel digital biomarker or endpoint is being evaluated, can be used physiological signals, for example, things like electrodermal activity or galvanic skin response, photoplethysmography, accelerometer data, and derive variety of features from these signals to objectively quantify pain measurements continuously, which would give us an ability to, understand the effectiveness and efficacy of a specific therapy, in patients with endometriosis. So with that, if we go to my last slide, the healthcare revolution is really at home, and we have seen because of the pandemic, trials are, going virtual. Delivery of care is being, virtual, and most of the health systems expect that 30 to 40% of care, which is traditionally delivered in the hospital setting, would move into patients' home in the next 3 to 5 years. And as that trend continues to grow, we see a massive opportunity in connecting drug development, digital health tools, and care delivery at scale. And this partnership, with the FDA and different, companies, pharma partners, health systems, all coming together towards understanding how digital health tools can be utilized in different care settings, in clinical trials, as well as healthcare settings, and coming up with regulatory, approaches and regulatory framework is extremely valuable for partners like us in making this a reality. So with that, I'll, thank everybody for the opportunity and happy to take any questions later.
Kuldeep, that was a fantastic presentation and powerful vision that you concluded with. It was also 3 really tremendous examples. It was exciting to see some examples around safety monitoring, so thinking beyond efficacy, if you like. I also really like the examples in the way that you sort of showcased, what we can do when we use multiple measures and multiple sensors. It's a great way to think about trying to overcome the challenge that we sometimes have with DHTs, which is an absence of context. When we have multiple measures, we can infer a lot. So terrific presentation. Thank you so much. And next, we'll move on to Neeta Sharma. So Neeta is the VP of Global Regulatory Affairs at Dexcom. And Neeta, I'll turn it over to you.
All right. Thank you so much. Can you hear me? Okay.
Yeah, we got you.
Okay, perfect. All right. Well, thank you so much for the opportunity to present Dexcom's perspective on the use of digital health technology in clinical studies. I'll focus on continuous glucose monitoring systems and glucose as a continuous variable that can really help us get more insights into therapeutic devices and therapies. So if you go to the next slide, before I get too detailed into what parameters we're talking about, I want to briefly get everyone acquainted to what is a CGM. A CGM is a device that's placed under the skin, and it measures interstitial fluid level continuously throughout the day and night. The glucose readings from this device are then sent to a smart device or a receiver as frequently as 5 minutes. And a CGM provides a lot more comprehensive information at the touch of your phone. In the United States at the moment, there are real-time CGM systems or intermittently scanned systems. And over the last year, CGM devices have become more easier to use, more accurate, and more accessible through the CMS and expanded coverage. The latest CGMs in the United States, called the iCGM, connect with multiple devices, including an automated insulin dosing system, thereby making diabetes management even more easier. And several of these devices also have remote monitoring capabilities that allow the data from CGM to be directly uploaded on cloud for better analytics, and they can be downloaded by a physician during a telehealth visit. They offer electronic health record integration as well. So overall, CGMs have been a great revolution in the glucose management, diabetes management paradigm over the last few years here. On the next slide, I want to briefly talk about the metrics that you actually get from a CGM. So historically or primarily, A1C has been the gold standard to really measure glucose control over time. However, a CGM provides a lot more metrics at a quick glance. They include mean glucose information, daily patterns of glucose excursions, time in range, and an estimation of A1C. All of these metrics are actionable, they are visual, and they help the patient and the healthcare professional better interpret the data and guide to treatment decisions. In the recent years, these metrics have increasingly become more standardized, and they do provide very useful clinical targets to enhance therapeutic decision-making. I want to focus on one specific metric for our talk this afternoon, and that is time in range. If you go to the next slide. So what is time in range? It's exactly as it sounds. It's the proportion of your time in a day that a person spends in their target glucose range. Per the international consensus guidelines, there are essentially 3 glucose ranges. And it really depends on how you manage your blood glucose, and target range for most people is between 70 and 180, and that's where really the HCPs target the treatment. And studies have shown that more time a CGM user spends in range, the less likely they are to develop certain complications. And without knowing the details of highs and lows on a daily basis, an HCP may prescribe a medication that does lower the blood glucose, but it may trigger hypoglycemia. And not all hypoglycemic events are asymptomatic, but if you look at time in range more closely, you'll realize that the pattern is very different. And overall, A1C is a good predictor, but sometimes they may still show that A1C was in control versus the user had several hypoglycemic events with the use of a certain drug. So as you can see, time in range can have a really big role in the management of diabetes and evolution of the therapies here. Let's look at the next slide and look at clinical implications of time in range. So emerging evidence has also correlated A1C very well to time in range. Essentially, the premise is that time in range is found to decrease serious life-threatening complications, enhance daily management of the diabetes, and then also improve the lives of people. In this specific study that I show here, Mayada et al. Have shown that association between time and range has evaluated through CGM, and then you can see the symptoms of peripheral neuropathy. Individuals with type 2D have lower incidence where they have higher time in range. So on the contrary, A1C was not found to be associated with peripheral neuropathy at all in this study. So as you can see on the next slide, we need to really move beyond A1C and utilize these digital health technologies and this glucose sensing methodology to really help understand the new disease management therapies. So A1C doesn't really document the highs and lows, and then people may have, as we noted on the previous slide, may even mask issues such as critically important hypoglycemia. So on this slide, you can see 3 patients had the same A1C, but their time in range was very different, thereby having an indirect impact on their therapy. Over the last 2 years, on the next slide, the international consensus for time in range has increased dramatically. Prior to 2017, time in range was reported in multiple ways, and it was almost impossible to compare one study with others. The first publication on CGM consensus was actually issued in 2007. And then subsequently in 2019, the international consensus for time in range recommended specific targets for type 1D and type 2D and also for pregnant women for certain targets in different glucose ranges. And then as late as December last year for 2023, ADA offered additional recommendations, including the use of integration of diabetes technology into care and also to expand broader access to CGM. And several esteemed organizations have issued joint consensus standards, really endorsing time in range as a really good metric that can be used for therapy management and also for overall diabetes management. So that takes us to our next slide, which is really how do we then use CGMs in clinical trials and research if it does provide all of these valuable tools and data analytics. So in recent years, CGMs have started to become more like a standardized method for glucose monitoring or almost as like a standard of care for many adults with type 1 diabetes. And also the CGM technology has evolved very rapidly in the last few years and in both accuracy and affordability. The CGM output has also been a source of research in exploration or as a secondary endpoint, but very recently we have started to see CGM as being used as in the assessment of primary endpoints. In this quick analysis here in 2021 that was published, there was an increase in CGM usage in clinical trials, demonstrating its popularity as a very useful digital health tool in clinical studies. Tying it back to our time and range discussion, if you go to the next slide, time and range has been validated as an outcome, as I mentioned, in clinical trials to complement A1C. And there have been now consensus guidelines given that a certain percentage points change in time and range is clinically meaningful for an individual participant, and there are detailed recommendations available. One of the first examples, a landmark study in range, was a 12-week multi-center randomized study that really compared the second-gen basal insulin analogs, glargine and insulin degludec in adults with type 1 diabetes to really show with a blinded CGM profile, and they actually showed that the two drugs, one of the newer drugs was more inferior, it was non-inferior to the therapy, and that they had comparable hypoglycemia in safety profiles, thereby really ushering a new era of use of CGM in the evaluation of drug trials. There are certain elements to keep in mind when we do select CGM devices for use in clinical studies. As I mentioned, there are many companies that make CGM devices available, and these CGM devices are slightly unique in their specifications, although they're all measuring glucose, but there are ranges such as the warm-up time, their wear period, their ease of use, etc. Their accuracy can be sometimes different, etc. So those elements should be kept in mind when picking a CGM for a clinical study use. Also certain brands of CGM, the staff and the users may have familiarity with certain CGM brands, but not all. So I think there has to be an element of training required as well when selecting a CGM. And lastly, I'll mention about the drug interference, because certain CGMs based on their chemistry and their mode of operations are prone to certain drug interferences. And those have to be kept in mind depending on the therapy you're doing a clinical study for, to really keep in mind whether they may have a confounding effect. And then lastly, I'll speak about the incorporation of CGM devices. Sometimes they can be used in a blinded mode, such as the user does not really have access to their CGM information. So you can download the data in retrospectively and analyze the time and range metrics that really allows for a more fair and balanced comparison of the therapy. So again, considering the heterogeneity of the systems that are available and how fast they are evolving, we recommend choosing the right CGM system is super critical. The next slide, here's another example of the SURPASS study, where I wanted to illustrate how time and range was utilized. So in this study, there was a sub-study done where a subpopulation of 243 participants wore a CGM for 7 to 10 days at baseline, and then 24 weeks and 52 weeks to evaluate the effect of tirzepatide compared to insulin degludec on time in hyper and hypoglycemic range. And in this CGM sub-study, they achieved the primary and secondary endpoints, and the CGM data showed that tirzepatide helped participants have less variability in their blood glucose levels throughout the day, thereby having a better time in range compared to the normal glucose ranges. I'm going to move to the next slide, which is really to just touch on some of the challenges. While you've seen incorporating CGM into clinical trials presents unique opportunities, it does have some challenges. However, there are challenges that can be overcome easily with some operational considerations, such as I mentioned, access to CGM devices, familiarity or lack of familiarity thereof by certain subjects, demands on patients and sites that this might put through, and then securing the clinical data or the data that's collected from these subjects. It has to be kept in mind. Certain participants may find a CGM inconvenient, or they may experience some discomfort, so that has to be kept in mind as well. And as I mentioned, the integrity of CGM data, it has to be kept in mind, so you can maintain the data strictly and just like you would maintain any other conventional data such as blood samples, etc. So in my conclusion, I would close by saying CGMs are a very powerful tool, and they have been utilized as a new and emerging digital health tool. They provide many benefits over a static blood glucose measurement or A1C, such as being continuous, real-time, accurate, and then combined with their remote monitoring capabilities and health record integrations, they're very friendly towards utilization in a clinical study. We have seen today how this continuous stream of data can actually be translated into very useful endpoints such as time in range. Time in range has been corroborated very well as a complement to A1C. As such, it remains a very valuable metric in clinical studies. And then by careful consideration should be given to selection of CGM devices. And last, I'll say that there's emerging consensus across the globe on using time in range, and a lot of effort has been put in on standardization of reporting across several scientific communities. With that, I will close and turn it back to the moderator.
Nita, that was absolutely fantastic. Thank you so much. And, you know, how compelling an example of how DHTs promise to make the previously invisible visible and allow us to better care for people. So thank you so much. And now we'll move on to our final presentation. So our final presenter is Dr. Dina Katabi, who's the co-founder of Emerald Innovations. So Dina, I'll turn it over to you.
Thank you very much. Hello, everyone. Today I'm going to tell you about this new technology, Emerald. And as you can see from my title slide, it says from wearables to invisible. So Emerald belongs to a new class of technologies where we can collect physiological signals continuously from people without asking them to wear any devices on their body. Next slide, please. So for the person who is moving the slide, could you click 3 clicks so that you can see the full slide with a red line at the bottom of the slide. So in this first slide, I want to emphasize the importance of data-driven drug development. Of course, drug development is always in our clinical trial, we are collecting data, but we want to strengthen this data-driven approach to drug development by having continuous clinical data from patients' homes. And not only have the data, but also be able to develop objective and sensitive biomarkers for difficult diseases. If you think about, for example, neurological diseases, in many cases we have PROs, we have scales, and these can be highly subjective. So if we have clinical data continuously from patients' home, particularly if that new data is powerful enough to allow us to provide a new objective and sensitive biomarkers, then it can help drug development drastically. Now, at the bottom of the slide, you see in red, the question really is how do we collect continuous clinical data from patients' homes while at the same time understand how to collect data from the sick and the old, because we are here trying to collect data from people who have Alzheimer's, from people who have Parkinson's, from people who have ALS. These are not the people for whom it's easy to wear devices and to remember to charge them and to have engagement issues with the devices. So how can we do that? Can you please move to the next slide? So on this slide, you see actually how we collect this data in patients' homes. So you see in the middle of this device what looks like a Wi-Fi box. So this is not just a Wi-Fi box. This is the Emerald box. So if you click next, it should show the word Emerald. So this is an Emerald box that looks very much like your Wi-Fi box at home. But the difference is that it sits in the background of the home and uses machine learning and AI to analyze the signal in the environment. And from that analysis with neural network, it can get sleep and sleep stages. It can get gait measurements, balance. It can get the breathing, heartbeat. It can know when somebody is scratching. And it can do all of that without asking people to wear sensors on their body, completely passively, while people going about their life as usual. So can you please click again and move to the next slide? So click to the next slide. So on the next slide, you should see a person standing in the middle of the office, and you see a big arrow pointing at the wall. So our device, the Emerald device, is not even in this room. We are going to monitor the movement of this person through the wall by by using the fact that when he moves, he changes the electromagnetic waves around him. You see a small inset on the side where you see a red dot. So this shows where the device thinks this person is standing right now. Click, please. So as we see now after the click that the video is playing and the person is moving. You should see the red dot moving with him. And remember, this person does not have wearables on him. It's just purely based on how his movement is changing the electromagnetic waves around him. And we are able to capture it through the wall from the office next door. Next, please. You see that whatever measurement that we collect, we compare to the gold standard. In the case of movement, the gold standard is something called the vital motion capture rule. And we show that the measure is highly accurate comparison to the gold standard. Now, of course, the difference is that the gold standard requires putting sensors and also changing the environment, putting infrared cameras all over the place on the ceiling. And here we can get this with completely passively without asking people to wear sensors. Next slide, please. So once you have the ability to track how people are moving in their environment, not only you can get their gait speed, which we know is important for a variety of diseases like Parkinson's, multiple sclerosis, etc. But also you can start talking about behavioral symptoms. So you can see, like, for example, if the person is like just very sedentary, sitting all of the day on a couch, for example. Or you can start like asking questions like how often the person enters to the bathroom or the kitchen and start understanding their behavior. Next, please. We also can monitor sleep stages. So as the person go to sleep, their brain waves change and they enter different stages: awake, light sleep, deep sleep, REM, or rapid eye movement. These sleep stages are very much underlying any sleep disorder, but also they are very important for almost all diseases. Sleep is really a platform for the vast majority of diseases. So just to give you examples, like in Alzheimer's, for example, the deep sleep or the slow waves that typically happen during deep sleep are impacted. In depression, rapid eye movements typically tend to change and happens too early during the sleep. So understanding these sleep stages is very essential for a variety of diseases. Next slide. So today, if you want to get sleep staging accurately, you send someone to a sleep lab and they put sensors on his body and his head, like you see on the slide with the person with all of these electrodes on his body. So they ask him to sleep like this. Of course, apart from the fact that this person is sleeping in a sleep lab, not in their normal bedroom, of course, with all of these sensors on his head and body, it's very hard to sleep normally. So let me show you how we collect sleep stages with our device. Next slide. So you see this white device is our device, transmits very low power wireless signal, analyzes the reflection using AI. And it generates what's called a hypnogram. So this graph that you see on the slide that shows different sleep stages: awake, REM, light sleep, deep sleep. This is something called a hypnogram. When you send someone to a sleep lab, they generate a hypnogram for him or her. And for every 30 seconds of sleep, it shows which sleep stage this person is in at that point in time. So we can generate that in the person's own home, as you can see, without putting any sensors on their head or body. They are sleeping in their bed, they are comfortable. We can do it every night. And it is highly accurate. Next slide. So the slide lists just a few publications that we have that look at the accuracy of our sleep staging and shows that it is highly accurate in comparison to the gold standard and in comparison also to FDA-approved devices for monitoring sleep. So next slide. So on this slide, you should be seeing a person who's sitting like you guys all, and he's reading. But what you see also is his inhales, exhales, his breathing. We ask him to hold his breath And you see the signal stays at a steady level because he exhaled, he did not inhale. Next click. So as I mentioned, for anything that we measure, we compare to the gold standard. In the case of breathing, the gold standard comes from FDA-approved breathing belts that the person has to wear on their chest or abdomen. And we can show in peer-reviewed publication that our measure is highly accurate. In fact, not only it's very highly accurate, in fact, both for sleep and breathing, we can extract these measures even when the person is sleeping with a bed partner. So even if there is somebody else sleeping in the same bed, we can still extract the sleep and the breathing of the participant in the study. Next. So on this slide, you see like a variety of images that represent different diseases. One of the things that I wanted to emphasize is that the Emerald device is already used in a variety of clinical trials as an exploratory endpoint. So we are in exploratory endpoint in trials including trials that have drugs in them. This shows just a subset of the trial or the type of diseases that we are in. Alzheimer's, Parkinson's, FSHD, Crohn's, COVID, atopic dermatitis, and there are a few others also that we have our devices collecting data and characterizing both the disease and the impact of a particular therapy. Next slide. So on this slide, I'm going to show you a video of the device. So if you click next, it will activate the video. So you should see the white box in the background. You see the radio signal. So it operates completely passively, just using the radio signal in the environment. Here you see a Parkinson patient as he moves. You see that movement as collected by the device on the floor map. Next slide. So I'm going to talk now in a bit more detail about clinical trials and some of the results from clinical trials that I can share with you. Of course, some of the clinical trials are under CDA, so I cannot share, but I can share a bit of results about Parkinson's disease. And this has appeared in our recent paper in Science Translational Medicine last September. Next slide. So in this trial, this is an observational trial with 50 participants. 2/3 of them have Parkinson's and 1/3 are control individuals, don't have Parkinson's. And everyone is monitored for 1 year. And next, please. So we collected a large amount of measurements, and I'm going to focus particularly on the gait measurement here that we collected, and we collected about 200,000 gait measurements in the home. Like basically, as people move in their homes, we can collect the gait speed of those individuals without asking them again to wear devices or do anything specific. Next slide. So the first thing that we did is to compare the gait speed that we get from our Emerald device to the gold standard. And in Parkinson, the gold standard is a scale that is called the MDS-UPDRS. Next, please. So you should see on the slide a figure that shows on the x-axis MDS-UPDRS part 3, which is the gold standard. And on the y-axis is the gait speed as selected by our device. And each dot on this graph is the average gait speed of one participant, one Parkinson patient. And you can see that there is a very high correlation between the MDS-UPDRS, which is the gold standard for assessing the severity of Parkinson's, and our device gait speed as measured passively at home. Next. So now you should see on the same slide also, on the x-axis, the MDS-UPDRS all parts, and on the y-axis is again our gait speed. And here again, what you see is that there is also still very high correlation. So on this slide, you see two figures that focus on two aspects of the MDS-UPDRS. One is part 3, which is the motion part, and the other one is all of MDS-UPDRS collectively together. And in both cases, you can see that the completely passive measurements that we get from the home are highly correlated with the MDS-UPDRS. So this first result shows you that what we measure in patients' own homes completely passively is very well grounded in the gold standard for assessing Parkinson's disease. However, one major problem of the MDS-UPDRS is that it is noisy, because basically it's a scale that has so many questions and it depends on patients, basically their subjectivity, as well as the subjectivity of the person who's running the test and how the data is collected. So because of that, there is a significant amount of noise in the MDS-UPDRS. So typically when you are running a clinical trial with Parkinson's disease, if you are trying to get statistical significance on the progression of the disease, then it will take you years before the MDS-UPDRS is stable enough to give you statistical significance. Not only that, but also you need something like 500 individuals for 5 years, for example, like what the Michael J. Fox did in their clinical trial. So what I'm going to look at next is a progression marker for Parkinson using our gait speed. Next slide. So on this slide, you should be seeing two graphs next to each other. One graph is from control individual, and another graph is from a patient with Parkinson's disease. If you look on the x-axis, you will see numbers from 0 to 12. So this is the number of months. So these individuals are monitored for 1 year. So what you are seeing here is how their gait speed is changing as a function of time. So you see that both the control, the healthy participant, and the Parkinson patient, in both cases, their gait speed is declining. And this is understandable because you are talking about people in their 80s. These are old people. We all, as we get old, the more old we get, the more we slow down. However, the thing that should be very clear to you is that people who have Parkinson's disease or this person who has Parkinson's disease is slowing down much faster than the age-matched control. So these are two people, but let's look across the whole cohort. Next, please. So now you should see on the right side of the slide the same thing but across the whole cohort. So you see the blue line is the decline in gait speed for the control, and the red line is the decline in gait speed for the Parkinson patients. And this is a linear mixed effect model. So you can clearly see that the Parkinson patients slow down over the same period of time twice as much almost. So this is very interesting because, next please, so you should see a banner at the top of the slide that says gait decline is statistically robust progression marker, and it shows you a p-value that is 0.04. So this is very interesting because for the first time, we can see that we have actually a progression marker that within a year is giving us statistical significance. And I don't know if any other progression marker that we have today that was able to show statistical significance in Parkinson within just 1 year, particularly with such a small sample size of only 50 individuals. So the implication of that is very important for clinical trials in diseases like Parkinson's. Today, as I said, you have to wait for a very long time. You have to have a large trial size, a large sample size of several hundred participants, if not thousands, to get statistical significance. But we can get it very in short period with a small sample size. Next slide, please. So on this slide, we are going to compare the sample size that is needed if you are to achieve statistical significance using MDS-UPDRS versus statistical significance using the Emerald device. Next, please. So you should see now a graph that on the x-axis, it says the reduction in disease progression due to new drugs. So this is the effect size of your drug. So when it shows 30%, it means you have a drug that has an effect size of 30%. Now, if you look at this graph, this is the number of participants in the clinical trial that you need if you want to achieve statistical significance on the MDS-UPDRS Part 3 within 1 year. And you can see very clearly that we are talking about several thousand patients. And in fact, if your drug has only 30% effect size, then you are talking more than 10,000 participants to achieve statistical significance on MDS-UPDRS within 1 year. Next, please. So now you should see on the same graph the amount or the number of participants that are necessary for a similar clinical trial, but using the Emerald device to achieve statistical significance within 1 year. And as you can see, the number of participants is much, much smaller. So effectively, what this means is that we can reduce the size of huge clinical trials that are needed today for Parkinson's disease to achieve statistical significance within 1 year. Or you can look at it in a different way. We can reduce the time from many years to just 1 year. And in both cases, it reduces the complexity, it reduces the time to market, it reduces the cost of clinical trials, and provides initial information for the pharmaceutical companies whether their drug is on the right path or not. Next, please. So now we are going to move to a completely different disease area. We are going to move and talk a bit about itch or pruritus. And for people who are not familiar with itch and how it is assessed today in clinical trials, so the way today we look at itch in clinical trial, the endpoint is typically something called the NRS, which is basically you ask your patient on a scale of 0 to 10, where 0 is no itch at all, and 10 is the maximum possible itch. So you ask them to rank their itch on that scale. As you can guess, this is very, very subjective because like my 5 can be completely different from your 5. So you are really like asking for a huge amount of subjectivity using such method. So what we wanted to do is to provide an objective way for assessing itch by assessing nocturnal scratching completely passively using wireless signal. Next slide. Next, it should show today pharma asked patients to rate their itch on a scale of 0 to 10, as I mentioned earlier. So next slide. Now you see on this slide, the one way to measure scratching at night is to use camera in patient homes. And in fact, we did this so that we have ground truth. So we put cameras in people's infrared cameras in people's bedrooms, and we collected videos, and we had people looking at these video footage, and whenever somebody scratches, they label this video as this particular instance is scratch. Next. So on the bottom of the slide, you should see the alternative approach that we are advocating, which is we put a wireless device in the patient's home, and we use the radio signal, and automatically using machine learning, we can tell you when a person is scratching and when they are not. Next slide, please. So now I'm going to show you the accuracy of identifying nocturnal scratching using the Emerald wireless device versus using videos that are annotated by a human observer that check whenever there is scratching. Next. So you should see now this graph that shows on the x-axis the human annotated videos, on the y-axis the Emerald prediction of scratching. So here what we are showing, this graph is showing the scratching time per hour. So the patient, as we said, we put in their bedroom both an infrared camera and had people look at exactly when they scratch. And we also put this radio device that we have with machine learning. And instead, so the way we measure things is by saying, what is the average amount of scratching per hour? So if you see like a number like 6 on the x-axis, it means 6 minutes on average every hour are spent fully scratching. So what you see here very clearly is that the Emerald device is accurately tracking the infrared cameras with the human annotation. But the difference is that you don't really need to collect videos in people's bedroom. You don't need to have people looking at those videos and you can achieve that accuracy just from radio signals. Next, please. So you see the R, which is the correlation, is 95%. The p-value is small, which means that it is trusted. Next, you see on the right side of the slide the ROC curve with the AUC of 97%, and you see that it has high specificity and sensitivity. Next slide, please. So this is the last slide. It says emerald from wearable to invisible. So really what we have here is a technology that we are talking about that belongs to a new class of technologies that are invisible in the sense that they disappear into the environment. They allow patients to continue to live their life normally while we are collecting clinical data. Of course, anything and any operation that we do and any data that we collect is according to IOB and informed consent. I'll stop here and we'll take questions later. Thank you.
Dina, absolutely fantastic. Terrific presentation on how we can think about DHTs beyond the context of a wearable, and also a really terrific example of how we can use these tools to reduce sample size. So Kuldeep, Nita, Dina, tremendous session. Really appreciate your expertise and you joining us today. We don't have time for questions, I'm afraid. So Christina, I'm going to hand it straight to you for session 5.
Thanks, Jen. I really appreciate that. And thank you for our session 4 panelists for dealing with some of our technological disruption. We are going to be moving straight into session 5 on key priorities going forward. We're going to be asking our panelists to reflect on what we've heard over the past 2 days and think about how DHTs can be used to move the drug development field forward. So at this time, I would like to invite all of our panelists to come on screen. And we'll start with a couple of remarks from each panelist and then go ahead and get started with our discussion. So first up, I'd like to invite Yuga Zhao from the Michael J. Fox Foundation to say a couple of words.
Thanks, Christina. And my name is Yuga Zhao. I'm really excited and honored to be here on behalf of the Michael J. Fox Foundation and the Parkinson's community here today. I know several panelists mentioned earlier already a brief overview of Parkinson's disease, but maybe as a quick refresher, Parkinson's disease, or PD, is a chronic progressive neurological disorder that affects nearly 1 million people in the US. And it's one of the most prevalent brain disorders and one of the fastest growing in the US and globally. There is no cure for Parkinson's, and there is no treatment that can slow down disease progression. Current therapies only address disease symptoms. And so this is really the urgency that is driving our foundation's mission to find a cure, and is what was entrusted by us and charged with us by our founder, Michael. And so as a patient-centered research funding organization, the patient impact is really a primary lens by which we evaluate our investments. And it was really exciting to see so many of projects that we have funded or supported highlighted in yesterday and today's workshop. So I think really exciting to see the ongoing work that is developing there. And so I think in the past couple of years, the foundation really has brought a new focus towards dedicating funding, partnership resources, and a full-time staff position, that's me, to supporting a strategy-driven program to advance more sensitive and patient-centered measures for both a regulator audience as well as payer decision-making. And in hindsight, this is something that we should have done many, many years ago, because this lack of sensitive patient-centered measures is really one of the biggest roadblocks facing clinical development for treatments that can slow or stop disease progression of Parkinson's. And something that I want to remind everyone as we talk through this panel is that the data that we collect with all of these different measurement tools comes from and belongs to the patient community. And that is something that is really, really important and central to our mission and goals. So keeping things short, I know we're running a little bit delayed on time. Maybe I just wanted to share a quick anecdote from a dear friend of the foundation, whose name is Gary, who is a member of our patient council and advisor to our endpoints program. This is around the gold standard measure that's being used in research for BD, the MDS-UPDRS, which is something that Dr. Kadabi mentioned earlier. So Gary, in a single day, he goes to 2 different research centers for a clinical trial visit. He gets assessed by 2 different clinicians, and he gets 2 different MDS-UPDRS scores. 2 different MDS-UPDRS scores. And talking with Gary, this is really confusing and frustrating because, he wants to know, why are his scores different? What does that difference really mean? And most importantly, how is he supposed to interpret the scores and their differences in terms of his Parkinson's disease status? And so I think this really clearly points to the need for better measures in Parkinson's. And I'm pretty excited about the potential that digital health technologies can be as a path forward here to developing better measures for patients with Parkinson's. So maybe I'll pause there and pass it back to you, Christina.
Thank you so much. And we are going to have a little extra time. We're going to spend a little more time on the session than was originally scheduled. So I don't want y'all to feel like you need to rush through your opening remarks. Next, I'd like to hear from Danielle French from Janssen Pharmaceuticals.
Great. Thanks so much, Christina. And again, thank you to the organizers for organizing such a insightful 2 days of meetings and discussions. And thank you for allowing me to participate. As Christina mentioned, I'm joining you from Janssen, where we've placed a high priority on developing, validating, and verifying, along with leveraging digital health technologies to support patient-centric drug development and review. Within Janssen, I sit within the regulatory policy and intelligence team. So just want to mention that that's kind of the angle or the perspective that I bring to the table today. I think as we were preparing for the session today, a couple of things that we agreed upon kind of as a panel was that we'd focus on the future, not the current state, thinking about how we can think about opportunities for transformational change versus kind of incremental change. And I think we've heard a lot of that throughout the course of the discussion so far. But then also to think about ways in which FDA might assist with regulatory clarity. And I think given the perspective that I come to the table with, I'd like to highlight a few things that come to mind for me. Before I do that, I just want to quickly mention that I think one of the most important, or one of the key themes that has been sticking out to me as far as the discussion yesterday and today was patient-centric approach. And how do we make sure that as we're thinking about leveraging digital health technologies in the context of drug development and review, we're starting with that patient perspective in mind. And so with that, I was thinking about, there's a lot of work that's already been done at FDA focused on incorporating the patient's perspective into drug development. So how do we connect all of that important, very time-consuming work that FDA has invested in advancing patient experience data and drug development review, and connecting it to the work that's being done to leverage digital health technologies. And I think that's especially important as we're thinking about all the stakeholders that are playing a role in leveraging digital health technologies in drug development and review, because some of them don't necessarily have the regulatory perspective or know that other body of work exists. So I think that's just an important comment I've been kind of coming back to as I hear everyone reflecting upon patient-centric approach. Again, I think maybe during the question answer session portion of this session, I'll highlight more of the areas where we see value in additional regulatory clarity, but just want to, first of all, thank FDA for everything they've done so far. It's been tremendous work. The framework came out very recently and lays out kind of what they've done so far, but also a whole body of work that's yet to come. And I think has been emphasized that this is just the first among many public meetings that'll be held focused on this topic. But things that come to mind as far as regulatory clarity include things like helping stakeholders better understand when their DHT is considered a device. And so I'll elaborate more on that as we go. Also considerations for when software updates or technology evolves, or perhaps technology becomes obsolete in the course of a clinical trial when that technology is being leveraged. How do we develop, or how do we follow approaches that best allow us to tackle those issues? And then the last thing that I'll quickly mention is, I think it's also important for us to think about our existing pathways that allow us to verify and validate the tools that we'd like to use in the context of our clinical trials. So whether that be in the context of an IND or drug development tool pathways, and thinking about ways that we can continue to improve those pathways so that we can best streamline the ability to bring these technologies forward in clinical trials and really support that patient-centric approach. So I'll stop there, but happy to elaborate as we get into the Q&A.
Thanks so much, Daniel. I'm sorry, Danielle. And then next up is Rebecca Nebel from Pharma.
Hi, Hi, good afternoon. First off, also want to thank Duke Margolis and the FDA for the invitation to participate in this important and timely workshop. For a little bit of background, PhRMA is a voluntary nonprofit association that represents the country's leading biopharmaceutical research and biotechnology companies, which are devoted to discovering and developing medicines that enable patients to live healthier and more productive lives. PhRMA is committed to advancing the regulatory environment on considerations for the use of digital health technologies in drug development. I think we've heard throughout yesterday and today the use of DHTs is enabling new opportunities to modernize healthcare, empower patients to make informed decisions about their health. They also hold great promise across the clinical trial ecosystem through their use in trial design, trial operation, by enabling remote data acquisition and contributing to the ability to conduct remote study visits. They may also reduce barriers, and we had a session yesterday about increasing access to clinical trials for underrepresented populations and geographically dispersed populations, and some considerations that we talked about yesterday that are needed to try and expand trial access while trying to expand the use of DHTs. While the integration of DHTs is occurring, there is an opportunity to expedite and increase the use of DHTs throughout the medical product lifecycle by addressing regulatory uncertainty. There's a clear need for continued guidance from the FDA on acceptable approaches to facilitate the use of these important tools. I want to echo my fellow panelists in commending the FDA's commitment to supporting use of DHT-generated data for regulatory decision-making, including draft guidances that have come out, specifically, the one on DHTs from the end of 2020, and the digital health commitments that are outlined in PDUFA 7, the framework that was released last week, publishing additional guidances on the use of DHT-based measurements, convening meetings such as this one to gather input from stakeholders and ongoing issues, conducting demonstration projects on methodologies to evaluate DHTs, and also enhancing internal systems and expanding capacity to receive and review DHT-derived data in a consistent manner. We have been talking about the large amount of volume that these tools can generate. So PhRMA looks forward to continued engagement across stakeholders to support the work needed to utilize DHTs in clinical trials to their full potential. I've been really encouraged by the discussions and case studies that have been presented during this workshop, and believe the DHTs in clinical investigations will continue to positively impact healthcare in the clinical research setting. So just thank you again, and I'll turn it back to you. Look forward to some more discussion in our Q&A.
Thank you so much. And Leonard Sachs from FDA.
Thanks very much, Christina. So I'm going to make my remarks very short because I had an opportunity at the beginning of the conference, which nobody else did. But I first want to say that from FDA's point of view, we're part of this learning community, and I think we don't know all the answers and nobody else does, but opportunities like this for everyone to get together are critical for us to advance the field. I think perhaps the take-home message for me from this meeting has been how much progress we've already made and what enormous abilities we have using different range of different technologies to measure very specific physiological phenomena. We've heard the range of applications, from neurological applications, glucose monitoring, cardiovascular monitoring, even depression monitoring. And I think it certainly opens the horizon for a lot of other indications which we haven't heard about, partly because perhaps we don't know about them, partly because they haven't been investigated yet. But I do want to mention that at the beginning, when I opened the conference, I shared a slide on the types of endpoints that are used to serve clinical trials. And there are a lot of unserved areas as well, which I think we need to sort of talk about. Things like rheumatological diseases, things about pain, measures of depression, measures of seizures, measures of abnormal movements. So I think the potential for digital health technologies is enormous, particularly in areas where we've had difficulty and we've relied on patient reports or caregivers reports. So I think this has been an enormously encouraging meeting in terms of the capabilities. I think it was also very helpful to hear some of the tremendous technological sort of progress that's been made by manufacturers. And not least, I wanted to echo the theme about patient involvement. I think something that we heard both from patient representatives and very much so from developers is the importance of being able to include the patients and include their curiosity in the clinical trial. I think that makes patients active partners as much as we are researchers. And that's an opportunity we haven't had in traditional trials. So I think those are some of the big messages that I've taken back from this meeting. But again, I want to open this as an opportunity for a little bit of frank discussion between our partners, particularly focused on ways in which FDA can help advance the field. So I'll turn it back to you there, Christina.
Thank you so much. And it's actually a perfect setup to my first question, which is really, where do we see the big opportunities for DHTs? And how can we build the trust so that the patients are going to be excited to jump with us into those big opportunities? And I am going to start off with Yuga.
Thanks, Christina. Yeah, I think this is music to my ears, hearing the focus on patient-centered drug development, the role of patient engagement, and how that is being highlighted and built into studies. I think one example maybe that I want to highlight is in Parkinson's. We are working together with the Critical Path Consortia to generate this consensus conceptual model for early Parkinson's. And this is actually stemming out of a problem that we have, where there's actually 3 different conceptual models for early Parkinson's, which is kind of a great problem to have, that there's a lot of qualitative research that has been done to understand what is important, what matters to patients in this population. But this is really hard for groups to build on, because there's so many different models that are out there existing. So something that we heard from stakeholders and regulators is that maybe if we were to unify and bring together what matters to patients in one unified model, this is something where, in the future, we can see groups building on this model, evolving this model, and then, instead of doing individual qualitative research themselves, can go to this model and say, okay, fine motor skills is something that matters to patients with Parkinson's, with early disease. This is something that we don't have existing great measures to, and so that now we can build a tool that will be really great at measuring this, instead of going over and starting that process over and over again, because we know that takes time, that's duplicative, and it really is a waste of resources and patients' time. And so what's really exciting about this project that we're building on with CPP is that it's involving patients with Parkinson's, it is involving industry, and it's bringing academic researchers all to the table. And a critical path really is also involved to help facilitate feedback from regulators on this project. And maybe an exciting note to bring here is that not only are some of the industry members that we invited didn't want to join, but we also have participation from some of the groups that are really the most advanced in building out their measurement tools in Parkinson's. And that's really exciting and encouraging. And it tells us, and it sends a signal to everyone that the individual competitive advantage that you might see from becoming a leading measure tool development, that they are still contributing, because the burden of proof for them to bring as an individual is much higher than what they would be achieved by building consensus as a collective. So when we think about building confidence in the use of these measurements for tools, I think the role of pre-competitive partnerships is really, really important. And I loved what Carrie said yesterday from Pfizer. I think these can be formal or they can be informal. They can just be, a conversation that starts. It doesn't have to be a formal collaboration. And so I really want to encourage tool developers to seek out these opportunities and really build and work collaboratively together to move the field forward.
Thanks so much. And I'm going to come back to that point about the pre-competitive space in a minute, but I do want to give some of the other people a chance to weigh in on some of where are the big opportunities for DHTs to make a big difference, either by themselves or to solve kind of a problems that traditional clinical trials may have. And then where do we need to build the confidence and the trust? Where is there uncertainty about this? Rebecca, I'd love to hear your thoughts on this topic.
Sure, yeah. So I think, the opportunities are, what we've been hearing throughout this workshop is, these tools can be used in a wide variety of disease and therapeutic areas. These tools can be more sensitive or more reliable than traditional tools. And as we've been talking about, there's an opportunity there as we're trying to identify these new outcome measurements, these new endpoints to incorporate the patient voice. And there's the opportunity to get a more complete, a more broader picture on how, diseases are, how these tools can measure patients across their daily lives rather than these snapshots that are, you get with a traditional clinical trial visit. And then using these tools in combination with other, other techniques to try and expand access to clinical trials across underserved communities, across geographical limitations. I think some of the areas, from the members that we represent and trying to build where we want to build confidence is, in having more regulatory certainty. And I know this is a developing area and, not these tools are new and sponsors and FDA need more, to gain more experience with these tools. So I do think the PDUFA 7 commitments are going to help with that, having more guidance, that takes into account, that provides some more concrete examples about the evidentiary support that's needed, across the spectrum of different tools and endpoints that are being used, whether the tool is approved and it's being used in the context of use that it was already approved, those for a specific endpoint may be very different from a new endpoint in a device that hasn't been cleared or approved, and the type of evidence that's needed for that. So just having some more examples, I think would be helpful for that. And the getting more experience, I think encouraging the flexibility to use these tools in an exploratory fashion first to gain some more experience, to then be able to leverage that and move that toward a primary, secondary.
And Danielle, I'd love to hear your thoughts on this too.
Thanks so much, Christina. I think those were great comments already made by the group. And I think there's been a lot of discussion over the last 2 days about, some of the areas that are, ripe for using DHTs in the near future. The other thing I just want to quickly emphasize too, kind of looking at blue sky and future, is that there are things that we maybe we can't even conceive of at the moment that will help us better or more earlier diagnose or develop endpoints that are earlier in the phase of the disease. And so I just want to emphasize that point, that there are things that we can't even think of at the moment, or patients might not even be able to articulate, that some of these digital health technologies will allow us to be able to better capture and hopefully develop therapies to treat in the future. I certainly echo Rebecca's comments around how do we build future confidence in the use of these tools moving forward. And I think, certainly from the industry perspective, we absolutely understand, that this is a learning experience, not just for industry, but also for FDA. And so I guess one thing that I've been thinking about too over the last day or so, especially as we think about the 4 additional meetings that will be had on digital health technologies, is how can we use these meetings to really tease through the areas we're having challenges, right? The areas where industry is not quite sure what to do, where FDA is not quite sure the best approach. How can we really work through those, maybe in a use case fashion, whether those are real or hypothetical, but really, tease through those issues to help inform both FDA, industry, and patient organizations who might be leveraging those technologies in the future. And then I'll just quickly mention that, I think from the industry perspective, a few things that I've heard on multiple occasions where additional regulatory clarity would be really helpful includes the piece that I mentioned, a DHT might be considered a device. And again, I think this is important because when we think about drug developers don't always have that device perspective, right? So fully recognizing FDA has guidance on, what constitutes a device, and there is a definition for that. It could be helpful in kind of thinking about how we lay out how someone would make the assessment as to whether their DHT is considered a device, and then whether the IDE requirements may apply. So some food for thought there. As I mentioned earlier, especially in circumstances where drug developers are using technologies that are developed by another entity, it's important for us to think about, what happens to software updates. And I think this is true for BYOD or, not. So thinking about software updates, technology evolution, or what happens if a technology becomes obsolete in the course of your clinical trial. And how do we address that? How do we make sure there's confidence in the data that we're collecting? I think the last thing that I'll just quickly mention as well that I kind of alluded to earlier is, how can we think about leveraging prior data in order to support expanded use of digital health technologies beyond their original context of use. And I know that the FDA guidance has alluded to the ability to leverage other types of data, whether that's collected by the tool developer, if that's not the same as the drug developer, but I think there's further room for discussion around that as well.
Thank you so much. And Leonard, you already touched upon this a little bit about where you see these opportunities, and would love to also hear, where are you hearing some of the common challenges or common questions and topics kind of coming up around this space?
I think perhaps one of the biggest challenges for us as an agency is satisfying our evidence standards. And, when new technologies come in, people don't have the experience, and we don't have sort of a calibration of how variable the measurements are in one arm or another, and what the effect sizes are. And I think it gets back to the issue of exploratory studies. We've heard an enormous amount of information today comparing DHT measurements with various others and so on and so forth. But what we haven't heard, and we may not get to hear, but what we haven't heard is how DHTs have borne up against measuring a drug effect. And when we think of the model of two arms and the need to satisfy our evidence standard, which is an adequate and well-controlled trial that can show that from which experts can reasonably conclude that the drug has an effect, I think it's important to think of showing that as well, showing that at the same time as you can distinguish between two arms when you add a new drug for Parkinson's disease or a new drug for diabetes, you're able to detect that difference using the DHTs as well as, or better than you can with traditional measures. I'm not sure how to implement that. This is a challenge. It needs funding, it needs dedication, but we know that clinical trials are underway around the world as we speak, and I'm sure there are many non-inferiority trials where arms are being compared for superiority, which provide opportunities to test our DHTs. And I would think that would be a very useful step in building confidence both among regulators and among industry. But, I'd be interested to hear what the rest of the panelists think, because obviously it's easier said than done.
I think great points, and would love to hear thoughts on that, and kind of related to that, and related to some of the comments that Danielle made in her opening remarks as well. This space is moving fast. Technology moves fast, and we talked about it in session 1 too. How do we think about verification and meeting those evidentiary standards in a space where the speed of regulations and the way this is done doesn't really match the speed of technological developments, and thinking through some of these issues around versioning, around updating, and things like that. Danielle, I'd be happy to start with you, or perhaps I know you touch on the space as well.
Yeah, I'm happy to just kind of kick us off here. I think, thinking through, how do you address when technology is changing so rapidly? And I think also this connects a little bit to the discussion that's been had, referring to gold standard throughout the last few days. And that, as different technologies, different endpoints are incorporated, that gold standard, if you will, may change. And that, I think that's something that is important to keep in mind as well. I think it's important for those leveraging digital health technologies to think about what are the impacts of these potential changes to the data that they're collecting, right? And making sure that it's not altering in any way the the results or the quality of the data that they're collecting. And so one thing I would encourage folks to think about is how perhaps developing a plan in advance for any potential changes to software, technology, et cetera, and how would you evaluate the impact of those changes to the data that you're collecting if it does in fact occur, through the course of your data collection.
I think that makes a lot of sense. And I know that on the CDRH side, there's been some thought around, kind of in a similar way around change control plans. Certainly an interesting way of thinking about it. Yvette, do you have any thoughts to add?
Yeah, maybe going back to Leonard's, your last point around, how can we really build the evidence that supports the use of these digital measures in measuring the effect size of in drug trials? And I think that really comes down to being able to, build a robust data set, even prior for these studies for being used as, primary, even secondary endpoints. And this is something that, we've as a funder have thought about, and something that our advisors have recommended. There's a lot of promising data, digital health technologies out there that have gone through, robust clinical validation. We just need to get them in interventional studies to really build that robust data set to see if they can be, used in further clinical trials for evidence generation, whether that's for a regulatory audience or for payers. And when we put forth an idea for a funding program to our advisors, one of them who has industry expertise was like, whoa, slow down, right? There are so many considerations that comes into incorporating a new measure, let alone a DHT into a clinical trial. There's a lot of technical, strategic, operational considerations that you have to think about. And so that's something that we're kind of working through in our head in terms of how can we incentivize, more data collection and generation around these novel measures that, can really support that. And so really curious to hear from my other panelists in terms of how you would think about building more data sets to support these novel measures, knowing that this is a very new space, very evolving space. And the only way that we can really learn together, regulators, industry, FDA, patient groups, is really to have some more data to show that.
And maybe sorry, go ahead.
No, go ahead.
I was just going to add that maybe one other piece to add to that too is I think there's been a lot of discussion throughout the course of the last 2 days around pre-competitive collaboration and how, bringing together those stakeholders can really support evidence generation for these technologies. The other thing to think about is there, platforms that are being built that, really support that pre-competitive collaboration. So if we can all do a better job of thinking about how we can work together in the spaces that make sense for that evidence generation, I think that makes sense as well.
And you read my mind, so this was perfect, because I was going to actually just go ahead and segue over to this pre-competitive space and thinking about this. And Leonard, I want to give you a chance to respond to the previous question as well. I know FDA has dealt many times with technology moving fast and regulation, having its own pace. But also, where can pre-competitive work help with this? And are there efforts that FDA is already involved in? Are there other efforts that you'd like to see?
Well, I think the pre-competitive space is critical, and there's a certain measure of, or at least there's a certain component of DHT development which is pre-competitive, and that's the measurement of a physiological feature and I think those measurements can be standardized, and consensuses can be reached on measurement of particular physiological features without any competitive issues arising. The competition obviously arises when you're testing a drug, and that's a different story. But I mean, for us to be able to validate a particular type of DHT for a particular type of measurement, whether it be exercise in cardiac disease or whether it be movement in Parkinson's disease, or whether it's quite frankly a measurement of psychiatric disease, depression compared to not. I think the standardization of measurements is something that's pre-competitive. And to me, a lot of work could be done there to get a consensus on the best type of measurements, and perhaps from there, we could move into the competitive space where we would look at things like exploratory endpoints embedded in clinical trials looking at existing drugs. So I think there is an enormous opportunity for pre-competitive collaboration. I think those are my initial thoughts on the pre-competitive space.
Thanks so much. Rebecca, do you have anything you'd like to add around where there's opportunities in the pre-competitive space to help move things?
Yeah, not too much to add from my other panelists. I would just say, in the pre-competitive space, and importantly, that we include all different stakeholders to this discussion, as we've been talking about throughout this workshop, and also echo, the standardization of measurements will also help, later down the road when, comparing across studies, comparing across, different applications, having that up front is key.
Thank you so much. And I'm going to actually come back to you real quick, because I think that, one of the things that we heard is, there are several other meetings planned as part of the PDUFA commitment, and would love to hear, where are there barriers or challenges that would be helpful to discuss more in this space. I'm going to go down the line on this as well. So Rebecca, can we start with you?
Yeah, I think I'd echo something I think Danielle mentioned, and we've heard throughout the workshop, is this, and I think everyone's probably mentioned this about, these gold standards, and moving away from some of these that have known limitations, and these DHTs can measure new aspects. We can measure endpoints that where a gold standard doesn't exist, and I think having some more examples and working through potentially some use cases of how we can verify and validate those measures would be really, really helpful.
Danielle?
I would echo Rebecca's comments there. Maybe reiterate some of the comments I made around kind of whether a DHT is considered a device. And then I think Matthew Diamond did a great job of talking about, when IDE may or may not apply. But, the slide did say generally. So I think, again, thinking about what are those cases that are not so black and white, and kind of working through those, I think could be really helpful. Maybe a few other areas for further discussion. I think the FDA recently announcing their new steering committee is excellent to hear. We're excited to see that steering committee carry out their work, but thinking through, what is the best way to engage the steering committee versus the center of excellence, and kind of working through that, I think could certainly be helpful for different stakeholders as well.
Thank you. And Eva?
Yeah, so maybe at the risk of maybe being a little too provocative for this audience, but just thinking an outstanding question that we have is around consensus across regulatory agencies. So as we started building out our funding and stakeholder engagement, we started partnering with other global advocacy organizations, because we know PD is a global disorder and sponsors are often global. And so there's really an outstanding question around, how can we build consensus around digital health technologies and digital measures, not just with the FDA, but also with other global regulatory agencies?
I wholeheartedly agree with that. I think certainly I've heard among different folks within industry that we're already starting to see differences in how health authorities are thinking about digital health technologies. And it does make it very difficult to carry out, global multi-regional clinical trials. So fully agree with that point.
I'll also third that, just underscoring the importance, moving towards convergence away from divergence, as this field is so new. Thinking about global harmonization is critical.
Well, thank you all so much. I really appreciate these thoughts. Leonard, you have been with us the whole 2 days, started us off, and so I'm going to let you end us off with any final thoughts you have, around the space.
Well, thank you very much, Christina. just in response to the last question before I move to closing the meeting, as we speak, colleagues of mine are engaged in ICH negotiations right now. And I think that's a very active area of interest. So as much as we can, FDA is very enthusiastic to promote global adoption and to promote standards that make it possible to perform multinational trials. Again, at this point, I think I'll turn to just some closing remarks. First of all, I wanted to thank everybody involved in this meeting. I wanted to give particular credit to our presenters who had to manage without their slides and to chime in on the telephone. I thought they did an exquisite job of presenting very fluently without any noticeable difference between normal presentations. I wanted to thank Duke Margolis in particular, excuse me, for conducting the meeting in a most professional and sort of provocative way. I think we got the information that we could. I thought there was a wonderful and diverse representation of constituencies in this meeting, both patients, developers, and manufacturers. So I thought this was indeed a very helpful start to a program that's very much in its infancy. I think everybody appreciates, as I've mentioned earlier, we're all early in the learning curve, and we're very anxious to figure out what the best next steps are, how, as you mentioned, Christina, how we should deal with these questions in future meetings, and what further regulatory advice or initiatives we should be undertaking to support the introduction of DHTs into clinical trials. So, as I say, in my view, this was a very successful first step. Again, thanks to all our participants and particularly to our audience. And at that point, I think at this point, I believe I'll be closing the meeting and thanking everybody, wishing them well, and looking forward to continuing the discussion. Christina, thank you.
Thank you, thank you. And thank you again to all of our panelists and everyone else during the meeting. I am going to hand it off just quickly to Marianne Hamilton-Lopez for just a quick final remark.
All right, thank you, Christina. And there's nothing more I can say to the eloquent remarks from Leonard, but just on behalf of Duke Margolis, we wanted to greatly thank our all of the audience, the moderators, the panelists, the presenters. Thank you all for being so willing to work with us through our technological difficulties today. And it's been a fantastic first start, and really appreciate the lessons learned and your willingness to stay with us and through some really complicated and complex issues over the course of 2 days. Thank you so much to the FDA planning team, especially Beth, Ryan, Leonard, and Deanne, and a very big thank you to our moderators, Jen and Lola. We really greatly appreciate all that you did to make this meeting successful. And then finally, a huge thank you to our Duke Margolis team, who has been working on this meeting for many, many weeks, and has put a lot of great thought into making it successful, and then was doing a lot behind the scenes today to make sure that we pulled everything off. So a great thank you to Aaron, Ethan, and Christina. So with that, enjoy the rest of your day. Concludes our event, and we look forward to future engagement.